Or
Let's go ahead and get started. Today is Tuesday, June 20th. We are continuing our exploration of the nervous system. We have a handful of assignments left, our Unit 13 and 14 reviews. One more Physio X because they're so much fun. One more graded for correctness assignment. I forgot to write that down. Again, that neuro review is all about structure, function, and location and how they go together. There is some redundancy on that, so make sure, so don't feel, get too confused if you see yourself writing down the same answer twice in a row. That's on purpose. And then a fun reflex activity that we'll do to finish things off. And then exams. So again, I think it's pretty straightforward what we've got going on from here on out. As I mentioned today, we are going to finish up our discussion of nervous physiology. We've done most of the heavy lifting on that. We have a little bit of new information to add, and a lot of it is really reorganization or rethinking about how we talked about things and adding some vocabulary to that. And then we've got some heavy lifting to do to catch up on our brain anatomy. So that is going to be the game plan for today. So if there aren't any questions, the exams. That I've cut you off at the pass, in case you would remember. Exams, I was able to get graded. Exams are graded and the scores have been posted. I will make the exams available today after class. As always, they'll be viewable for about 24 hours. And then that'll give you a chance to check and see if there's anything, any questions or concerns or comments you can ask me about. Come to my office hours, send me emails. And then if you are not able to view it during that time, then we will, you can always come to my office hours and meet on with me on that. So that is the game plan for that. So hopefully this will help you to prepare for the last exams. All righty. Any other questions? Excellent. All right. So as we have mentioned, we have been talking about a neuron, and we have now know how on the cell body and dendrites, it is able to produce a graded potential. It is a small variable signal that can be positive or negative depending on the types of channels that we open. And they produce small waves that travel small distances to our axon hillock. There in the axon hillock, we have our specializations of the axon, the axolemma beginning, and then we have that trigger zone where we're able to produce our first action potential. We now know how to propagate that action potential down an axon on both a myelinated and an unmyelinated axon. And as I mentioned in the previous class, that really only leaves us with one thing: how neurons communicate with whatever their target cell is. So we have these target cells that our neuron is going to communicate. And as we learned before, there is a special name, generic name we give for this specialized region of communication. And what is the special name we give to this specialized region of communication?
Neuromuscular junction.
synapse.
True, absolutely. The neuromuscular junction is one example of where a somatic motor neuron communicates with skeletal muscle cell. But the neuromuscular junction, remember, was just one of dozens, if not hundreds, of different types of specialized regions for communication. And what was the generic term we used for it? There we go, synapse. Absolutely. But you're absolutely right. The neuromuscular junction is where we were introduced to this, where our somatic motor neuron spoke with the target cell, that in that case was our skeletal muscle. So anywhere where a neuron communicates with its target cell, we call that a synapse. And just out of curiosity, anybody know what the most common type of target cell might be? What do you think most neurons talk to?
Other neurons?
Exactly. The most common target cell of a neuron is actually another neuron, which makes sense. If you remember, our most common functional type are interneurons. They're the ones in our central nervous system, making up about 90% of our neurons. They communicate between the sensory and the motor and between each other. So it's not surprising that our most common type of target cell is another neuron. Most neurons talk to other neurons. Excellent. So we have, actually, let's go ahead and erase that. So we know we have this region, specialized region of communication. And again, it's called a synapse. And as it turns out, there are actually two specific types, two main types, let's say. And we've actually talked about both of them. I know that may be somewhat surprising because we didn't use the term synapse at the time. But up to this point, when we've been talking about synapses, we've actually been, or so far when we've used the term synapse, we've been talking about what we call a chemical synapse. As we know, that chemical synapse has three components. Right? It has that synaptic end bulb, which, and again, since most neurons talk to other neurons, we can't just say it's a neural specialization anymore. So we can say the synaptic end bulb of the presynaptic cell. We have some type of specialization of the postsynaptic cell. So like, for instance, in our skeletal muscle, that was the motor end plate, where we had our invaginations, we had our cation channels. Since most neurons talk to other neurons, that would be the dendrites. There's specializations to the dendrites or something like that. And as we also know, the cells don't touch. There is the space in between. And what do we call the space in between?
Synaptic cleft.
There you go. The space that is in between them as well, right? Again, one of the keys to this chemical synapse is they do not touch. Instead, a chemical signal. And what's the generic term for that chemical signal again?
Transmitter.
Say again?
Neurotransmitter.
There you go. A neurotransmitter is released, spreads across that synaptic cleft, and opens some type of chemically channels, allowing ions to flow. And when we do that, we produce a current. And if that current is big enough, we get an action potential in the target cell or some other type of effect. Now, if you think about it, as we've talked about it, is there one-way or two-way communication with this type of system?
One way.
Yeah. One-way communication. And do all of these effects occur instantaneously?
No.
No. So again, we're talking relative here, but it is a relatively slow process. Again, we're as was pointed out before, we're talking about, you know, milliseconds here. So I mean, again, it's relatively slow, but there is that a bit to it. But as we've also talked about, the advantage of this is that it is modifiable. We have the ability to affect this. It's not always going to be the same. Even something that we think of like a reflex. We talked about the example before. I hit your knee with the hammer and your leg kicks out. And then I hit your knee with the hammer and your leg kicks out. And I could do that all day long and your leg's going to kick out. And then I put a sleeping baby in front of your foot. And when I hit your knee with a hammer, is your leg going to kick out?
No.
No, you have that ability to modify it. Same stimulus, different responses. It's a huge advantage of these chemical synapses. That's why it's not surprising that this is the most common type in the nervous system. It's because it has that ability to be modifiable. So again, our first major type of synapse is a chemical synapse. And that's primarily what we've been talking about up to this point when we use the term synapse. But technically, there is a second type of synapse. And that second type of synapse is what we call an electrical synapse. We didn't use this term before, but we did actually talk about it. An electrical synapse is when two cells, and again, normally these cells are something like neurons or other types of specialized cells. So let's just use it with two cells. Actually touch, are actually connected. And they're connected to each other by gap junctions. What was the protein that helps to form those fluid-filled pores in the cells to allow them to rapidly communicate to each other? Anyone remember?
Desmosomes?
Say again?
Desmosomes?
Remember, desmosome was another type of cell junction. You're right, it is a cell junction. But proteins that form the pores were connexons. Again, we've learned a million things in this class. It's okay if you don't remember that, but it is something that we did talk about. So we have those connexons that form a connecting pore between the two cells, right? So we have a fluid-filled pore that directly connects the cells. All right. There are some huge advantages to this. One of those advantages is that, of course, it is very fast. Instead of having to let sodium come into the synaptic end bulb after we open voltage-gated calcium channels and then cause the mechanism, all those proteins, to bring a chemical signal to the plasma membrane, to release it via exocytosis, to have it diffuse across the synaptic cleft, to bind to the target cell, to open some channel, to allow ions in, to produce some current, right? With gap junctions, if you have a big, huge flow of ions of sodium, that big, huge flow of sodium can move straight on through those holes to the next cell. So it is incredibly fast, right? We've used this term when we talked about it. These cells are hardwired together. Basically, what one cell does, the other does. So these electrical synapses allow the cells to work together as one. And if I have two cells, cell A and cell B, and cell A has a massive influx of sodium, it's going to flow to B. But what happens if cell B has a massive influx of sodium?
Flow to cell A.
Yeah. So notice another huge advantage of these electrical synapses is that it allows two-way communication. So if this system is so awesome, why is it that this is not commonly found in our nervous tissue? Again, notice I didn't say it's never found. It is found some in our nervous system, but as we also talked about, it's more common in things like cardiac muscle or skeletal muscle, things along those lines. Why is it that it isn't as commonly found in our nervous tissue if there's so many great advantages to it?
Because you want the action potential going one direction.
True, that could be part of it. We do want it to maintain a directionality. As we've learned, direction is very important. What's the other huge disadvantage?
It's not
Not
modified.
modifiable.
Yeah, so it does not maintain that directionality. And the big one, like we talked about, is it is not modifiable. It's like the light switch, hardwired. I could sit here all day long flipping my light switch, and all that's going to happen is my light here in the bedroom is going to turn on and off and on and off and on and off and on and off. There's never going to be a time where I flip that switch and the garage door goes open, or the garbage disposal goes on, or the back door opens, or any number of other things, right? And thankfully for sleeping babies, the same is not true for our reflexes. When you hit your knee, you don't kick that baby, unless I guess you want to kick the baby. But if you don't want to kick the baby, you can stop it because our chemical synapses are modifiable. Again, if you think about it, this really isn't new information. We already learned about gap junctions. We just never really gave them the term electrical synapse. So really, we're just adding a new vocabulary term to something we already understood. All right, so again, as I mentioned, most of our heavy lifting is done. But we're adding a little bit of layers, we're adding a little bit of vocabulary, making sure we understand this information. Two types of synapses. The first one we talked about, even though we didn't use the term electrical synapse, we definitely talked about electrical synapses, and that's when cells are actually connected, touching each other, held together by those gap junctions. Fast, two-way transmission, synchronizes many cells, and will be found in nervous tissue, but also remember we saw it a lot in muscle tissue as well. Chemical synapses, on the other hand, as we know, do not touch. There's the space in between. Our presynaptic cell releases that chemical signal. There's a little bit of a delay in getting that signal across. It only allows one-way transmission. But the key to this is that it can be modified. And we can get different results. One of the reasons we can get different results is because we can produce different types of signals. And the way we produce different types of signals is by opening different types of channels. Again, this is not new information. You guys already know this. If we open a sodium channel, what's most likely going to happen to that target cell?
Get more positive.
It's going to get more positive. If we open a chloride channel, what's going to happen to that target cell?
Get more negative.
It's going to get more negative, exactly, right? Notice we are going to produce some type of postsynaptic potential. Again, just adding vocabulary. Right? Postsynaptic, remember, is the cell after the synapse, or the target cell in the synapse, let's say it that way. So our postsynaptic is that. And as we already know, it's going to produce some type of graded potential. That can be positive or negative. That can be bigger or smaller. All right. So again, we haven't used the term postsynaptic potential before, but it isn't new information. We're just adding vocabulary to what we already know. And the same thing is true for this concept of excitatory or inhibitory. We have to explain that, but we'll get to that in a second. Again, not a new concept. We're just adding some vocabulary to what we are discussing. Now, here we see an example of a common synapse. Notice, again, most neurons communicate with other neurons, but the process is identical to what we learned in the muscular system. We have a neural action potential that travels down the axon to the synaptic end bulb, causing a big depolarization. That depolarization opens calcium channels, allowing calcium into the synaptic end bulb. Calcium entering the synaptic end bulb triggers the process by which vesicles are brought to the plasma membrane and release their contents via exocytosis, where it spreads across or diffuses across the synaptic cleft to bind to some chemically gated channel on the other side, opening those channels and allowing ions to flow in or out. And that movement of ions into or out of the cell produces a current, which we call, as we just mentioned, a postsynaptic potential. Again, absolutely, positively, literally no new information on this. One change in vocabulary, right? The postsynaptic potential. But other than that, this is the exact same process we saw at the neuromuscular junction. It's how all chemical synapses work. So I know I just went over it, but I'm not going to bother drawing it and writing it out and doing all of that again, because we already did that to exhaustion in the last section. Now, just because I didn't go over it again, does that not mean that this won't be an essay question on the next exam? On the next exam, can I ask you how a chemical synapse works?
Yes.
Yeah, absolutely, right? Because again, this is a cumulative class. You need to know this information. It was a huge part of the last section. So again, absolutely, that could be a potential essay question on the next exam. All righty. But you already know it. So that's easy breezy. That's the one you want to get on the next exam because you already know it. All right. Now, as we talked about, binding of a neurotransmitter to that target cell, that postsynaptic cell, is going to open ion channels. Ions flow in and out, and that produces some type of graded potential. And again, that graded potential we give a fancy name to. We call it the postsynaptic potential. No new information here, just a new vocabulary. However, what is slightly new, and again, it really is just layering on what we were talking about before, postsynaptic potentials can be depolarizing, right, or hyperpolarizing. Those are terms we've used before. But similar to that is excitatory and inhibitory. Now, let's switch back to the whiteboard and see what we mean by this. Here is our graph. Time, voltage, and there are, as we know, and we'll go ahead and put all of them on here, 5 important values, important voltages we need to know. Give them to me. Pick one.
Equilibrium potential of sodium.
Excellent. What's that one?
Positive 55.
66, close. Excellent. And you're right. Let's work our way down. I like this. From there, oh, okay. Never mind. We'll do that instead. Absolutely. Our resting membrane potential, which again, I'm going to abbreviate because we've been writing it so much, negative 70 millivolts. What's next?
The equilibrium potential of potassium,
Excellent, which is?
negative 90 millivolts.
Perfect. Yeah, I know. Millivolts, I haven't right. So yeah, you're right. Let's put millivolts up here so we remember that's what we're talking about. Excellent. 2 more.
Threshold at negative 60.
And one more that we learned. What's the most recent one we've added to the list?
Inactivation.
Close. I like that, but it isn't. So that's a great one. Absolutely right. What happens is we reach a peak of the action potential. We reach a peak of the action potential, which we said was what?
Positive 30.
Positive 30. That's a great point. It isn't that when the cell reaches positive 30 millivolts, the ball and chain wedges itself into the pore. It's not like a voltage gated channel where it's affected that way. It's more of a timing issue. They've actually, again, as I said, I studied the field of neuroscience, but I did it at a systemic level. There are some of these microanatomists who are studying these molecules at the molecular level. And again, it is research I would never want to do, but some of the stuff they do is remarkable. There are some researchers who have actually been able to go in and modify the length of that chain of the ball and chain by adding or removing proteins from it. And what they found is that the ball wedges into the pore just by random movements. So when they shortened the chain, the ball was closer to it and it would lock it more quickly. And with a longer chain, it would move around more randomly before it finally accidentally wedged itself in there. So it's really the timing. It isn't a voltage. It isn't like that when it hits positive 30, the light, the switch hits. It really is more of a timing issue. But you have absolutely the right idea. Because the channels inactivate, the voltage-gated sodium channels activate, because the potassium channels finally open, those two events occurring at about the same time causes the cell to peak at that plus 30. So it isn't a threshold the way threshold is. It's not like when you hit that, suddenly they inactivate. It's more the other way around. They inactivate, and because they inactivate, the signal stops at positive 30 millivolts. Excellent. All right. So here are the 5 major voltages that we need to know. And as you guys have kind of already pointed out, to get this cell to do work, the primary one we care about is this threshold. Because as we've talked about threshold, when we say the word threshold, really what we mean is this is the voltage where all voltage gated channels will open. And of course, sodium are the ones that happen first. And at the same token, then, since most target cells are neurons, this is basically the voltage where we get an action potential. Or where we can, let's say it this way, where we will produce an action potential. Excellent. Again, no new information here. This is all stuff we already know. We know the cell normally starts about negative 70 millivolts. And to stick with vocabulary we already know, we know that when we get more positive then resting membrane potential, we call that a depolarization. And when we get more negative than resting membrane potential, we call that a hyperpolarization. Again, no new information here. So here's where we, again, start layering stuff together. In our postsynaptic cell, right, our target cell, we, using a chemical synapse, oops, we open channels and allow ions to flow into or out of the cell. We produce that current. Graded potential, current, and again, now there's a new term we call for it, a postsynaptic potential. And we know, oh, didn't mean to do that. We know these postsynaptic potentials can go in one of two directions. We can make the cell more positive. And notice when we make the cell more positive, we make it more likely to reach threshold. Notice I didn't say it guarantees we're going to reach threshold. It just makes it more likely. And if it makes it more likely to reach threshold, we call it excitatory. So an excitatory postsynaptic potential makes the cell more positive and makes it more likely to reach threshold. Conversely then, an inhibitory postsynaptic potential makes the cell more negative. And if it makes it more negative, it is less likely to reach threshold. Now, why do I say more likely or less likely? Doesn't it either have, doesn't either have to get to threshold or not get to threshold?
Yeah, but you might not want it to get to threshold.
True, you're right. You might not want it to get to threshold, but here's the other thing as well. When we are if you remember, we talked about a single neuron could receive as many as 10,000 synapses on it. So it could be getting 10,000 pieces of information. So if one neuron produces one excitatory postsynaptic potential, do you think that neuron is going to fire an action potential as a result of that?
No.
No, not necessarily, especially if the other 9,999 are sending inhibitory signals, right? So each individual potential makes it more or less likely, but neurons are basically accountants and not like Twitter accountants. I mean, OnlyFans accountants. I mean, like real bean counting accountants, right? Where they are counting the number of positive and counting the number of negative inputs and then deciding whether or not they're going to fire an action potential. If 9,000 synapses are giving us excitatory postsynaptic potentials, and only 10,000 are saying inhibitory, well, then that cell is likely going to fire an action potential. But each individual postsynaptic potential can be either excitatory or inhibitory, make it more likely or less likely to reach threshold. And we can add one more piece to this puzzle. What ions, or let's say ion in this case, is most likely going to be used to produce an excitatory postsynaptic potential?
Sodium.
Sodium, absolutely. Remember, calcium can do it as well, but remember, calcium makes cells do wonky things, so it's not going to be as common. Whereas what ions, plural, are the ones that will most likely be used to produce an inhibitory postsynaptic potential?
Potassium.
Potassium and chloride.
Yeah, potassium and chloride. Perfect. There you go. Let's look at the pretty pictures. Notice here we have an example of a excitatory postsynaptic potential. We have a stimulus that causes a depolarization, gets it closer to threshold, making it more likely to reach threshold. But remember, it doesn't have to get there to be considered excitatory. A graded potential that gets you to, right, negative 61 millivolts is a pretty big excitatory postsynaptic potential, but you're not getting an action potential as a result of it. You're not reaching threshold, but it is still considered excitatory. And as we talked about, typically this is going to involve sodium channels. Conversely, an inhibitory postsynaptic potential causes hyperpolarization, makes the cell less likely to reach threshold, and typically that involves either potassium or chloride channels. So again, if you think about this, oh, go ahead, Brian, did you have a question?
Yeah, when I was doing the review last night and I was like learning about this, and I was trying to figure out, when it came to the excitatory, that's like, hey, I want to move my muscles, and you're thinking about it, it's like an active process kind of thing. What would make it do the inhibitory? Like, that's not something you would think about, or?
Well, okay, so, okay, let's think of it this way. If it is in terms of muscles, right? You touch something hot, and we'll actually talk about this later on, but this is a good example. If I touch something hot, what do I want to do?
Pull your hand away.
I want to pull my hand away. How do I do that?
Reflexes.
Well, true, okay, but let's be more specific. Of the muscles that we've talked about in this class, what muscle would be involved in flexing the elbow to pull you away from something harmful?
The specific?
Yeah.
Biceps brachia.
Biceps brachia, excellent. However, what muscle was allowing you to extend your hand to reach out and touch that candle in the first place?
Triceps brachia.
Right. When I grab an electric wire, what happens?
It contracts all of it.
Contracts all the muscles, so my arm doesn't go anywhere, and that's definitely a bad thing. So when I put my hand over something hot, I definitely want to excite my bicep brachia so I can pull away. But what else do I need to do?
Relax your tricep.
I need to inhibit my tricep to make sure that it's not contracting anymore so that I can pull it away.
Gotcha.
So that would be a perfect example of a situation where you would want to, and at the same time, be exciting one muscle and inhibiting another to make sure that I am able to do that protective motion of pulling away from a painful stimulus.
Awesome. Thank you. Yeah, I was trying to figure it out. Like, where would this be? Okay. Makes sense now.
Awesome. And again, there's lots of other examples. Absolutely. Right. So, but that's one of the most obvious and an easy one. And we'll actually draw that pathway. We'll actually see how that works. All right. Awesome. So there you go. Again, not a tremendous amount of new information, but we've added some layers of vocabulary to the things like postsynaptic potential and excitatory and all of that. As we also just finished talking about, typically one postsynaptic potential, or more specifically, one excitatory postsynaptic potential, isn't enough to generate an action potential. We haven't thought of it in those terms, but right, all you have to do is a little bit of math to figure that out. What did we say the average graded potential was?
2 millivolts.
Yeah, 2 millivolts. And how many do we need to get to threshold?
10.
Yeah. So if you think about it, even though we didn't emphasize it at the time, this is something we kind of already put out. Is it possible to get one big graded potential that is big enough by itself to reach threshold and fire an action potential? Absolutely. But that's typically not how it works. And one of the main reasons for that, as we talked about, is a single neuron can be receiving as much as 10,000 different pieces of information. It can have 10,000 synapses. So it could be receiving 10,000 postsynaptic potentials. And so what that neuron is going to do, like I said, it's an accountant. It adds them all together. As I talked about, if I have 9,000 excitatory and 1,000 inhibitory, then is this cell likely going to be firing an action potential or not?
Yes.
Yeah. What if, on the other hand, I've got 4,000 excitatory and 6,000 inhibitory?
Nope.
And again, if you think about it, we can take it one step further. This neuron could be getting 5,000 excitatory, 5,000 inhibitory inputs. A massive amount of information and signals are being received and movement of ions and all this stuff going on. And pretty much what's the net effect on that cell going to be?
Nothing.
Yeah, pretty much is. It's definitely not going to fire an action potential. But overall, it's really not going to have any kind of significant change on it at all. Right? Because just like dollars in your checking account, one going in basically counts the same as one going out. One inhibitory postsynaptic potential basically cancels one excitatory. So if you have 5,000 positive and 5,000 negative, you're pretty much right where you started.
Wouldn't the size of the postsynaptic potentials matter as well?
Yes, absolutely. So again, we're totally sky is bluing it here as well. Because if you think, because again, we could spend hours and hours talking about the synapse. It's one of my favorite things. When I was in graduate school, we had to do an oral exam as part of it. And the first hour and a half of my exam, we just talked about the synapse. It was a horrible, horrible experience. but there's so much to, but yes. So if you think about it, remember, our goal is to get a wave basically to this region, to the axon hillock. And just like when you're in a pool, if you produce two waves that are exactly the same size, a 2 millivolt wave right here on the cell body, and a 2 millivolt wave way back here at the furthest dendrite, are these two waves going to be the same size when they reach the axon hillock?
No.
No. So notice it's not truly even. If one of these was excitatory and one of these inhibitory, they wouldn't truly cancel themselves out. But again, in this class, the sky is blue. In its simplest form, it really is accounting. Again, it is like everything a little bit more complicated than that, right? Because yes, you make money and yes, you spend money. But every time you spend money, you don't spend the same amount. Every time you make money, you don't make the same amount. So every paycheck doesn't cancel out every grocery bill or vice versa, right? It is a little bit more complicated than that, but in its general principle, this is how it works. Now, we will talk a little bit about one way how it's difficult, because we do have this math issue. Where out here, we are producing graded potentials. And these graded potentials need to be added up at the axon hillock. Your book has this great analogy for this that I really, really like. They use the example of, all right, you've got a bathtub that you need to fill with water. All right. So your bathtub is upstairs on your second floor and your faucet is downstairs in the basement. And let's say, for instance, it takes 10 buckets of water to fill up our bathtub. That would mean I'd have to go down to the basement, fill up a bucket, walk up the 2 flights of stairs, dump it in the tub, go back and do that, and I'd have to do that 10 times. And if I did that 10 times, I could fill my bathtub, correct?
Yep.
Yeah. But here's the problem. You've lost the stopper for your bathtub. So if I walk down the 2 flights of stairs, fill my bucket, bring it back up, dump it in there, walk back down the 2 flights of stairs, fill my bucket, bring it back up the 2 flights of stairs. By the time I've gotten back to the bathtub, what's happened to my first bucket of water?
It's gone.
It's gone. So yes, I need to be able to get 10 buckets of water into this tub, but I have to either do that 10 buckets together close in space or close in time. Now, one of the ways I could do this is, instead of having my bathtub up on the top floor and my faucet down in the bottom, I could have them next to each other. And then there, me by myself, I could fill a bucket and dump it and 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 fill a bucket and dump it. And if I can do that fast enough, am I going to be able to fill that to the top before it all drains away? Well, yeah, if I do it fast enough, as long as I pour water in faster than it drains out, especially if I have 3 buckets and I'm constantly filling them while I'm dumping other ones. If I do it fast enough, I can get it filled, right? As long as I can get water into the tub faster than it drains out, right? Those normal drains are pretty small. My bucket's pretty big. So if I do it fast enough, I can get it in by myself. However, if I want to take a bath, I could also call up 10 of my closest friends and say, hey guys, why don't you come all over? I need you to help me fill my bathtub so I can take a bath. And we all fill our 10 buckets and we all dump our 10 buckets at the same time. If me and 10 of my friends all dump our buckets at the same time, can we fill the bathtub then?
Yeah.
Yeah. And so that's exactly what we're talking about. This concept is what we call spatial and temporal summation. Temporal, as the name indicates, is I do one neuron, so this is me filling it by the self, that fires many action potentials very close in time. When I fire by myself many action potentials close in time, as a result of that, I produce many postsynaptic potentials. And basically, this is similar to what we saw with twitches and wave summation. I'm able to add my graded potentials together to reach threshold. So me by myself firing many, many times can produce enough graded potentials where I'm able to add those graded potentials together and reach threshold. Or with spatial summation, I can have many neurons all in the same location. Or not the same location, but let's say all on the same target neuron. That's why this is spatial summation. They're all in the same location. And because they're all in the same location, when they all fire, we are able to reach threshold. So whether it's me by myself rapidly dumping buckets, or me and all my friends all dumping our buckets at once, we're able to fill that bathtub. We're able to reach threshold and produce our action potential. All right. Questions on that?
Nope.
All right, excellent. Last little bit we need to do for lecture today, and then like I said, we're gonna spend the rest of our time on our lab, is to talk about neurotransmitters. As I mentioned, over 100 different chemicals have been identified as neurotransmitters found in the body. What's important to remember about neurotransmitters, or typically we talk about neurotransmitters being excitatory or inhibitory, but it isn't actually the neurotransmitter itself that determines that. Let's talk about a great example. Here is neuron 1. Here is neuron 2. And both neurons happen to release acetylcholine. That's convenient because we know that acetylcholine has an effect on skeletal muscle. What is the effect that acetylcholine has on skeletal muscle? Is it excitatory or inhibitory?
Excitatory.
Yeah, it is excitatory. It is excitatory. It binds to a receptor found here on our skeletal muscle cell and is excitatory. So, by that same token, I can also sprinkle acetylcholine onto cardiac muscle. Any idea what happens if I sprinkle acetylcholine on cardiac muscle?
The opposite.
Exactly. Here, the muscle contracts. However, if we sprinkle acetylcholine on cardiac muscle, it actually is inhibitory in its effect. It actually slows the heart rate down. So how is it possible that acetylcholine can be both excitatory and inhibitory?
Based upon the muscle tissue it's acting on.
True, but what is it about the muscle tissue that makes the difference? Well, let's think about it. Go ahead, what were you gonna say?
I was going to say the channels that are attached to the.
Exactly. That acetylcholine binds to what type of channel in our skeletal muscle?
Chemically gated cation.
Yeah, it's a cation channel.
Cation.
And it's a cation channel, true, but what cation primarily flows through it?
Sodium.
Sodium. And we know sodium is responsible for exciting cells because sodium goes in. It turns out on cardiac muscle, acetylcholine binds to a completely different channel, a different type of channel. In this case, it turns out to be a chloride channel. So when you open up that chloride channel, what's going to happen?
Chloride's gonna rush in.
Chloride rushes in and it inhibits the cell. It slows the heart rate down. So notice it's the exact same chemical, acetylcholine. So often when we talk about neurotransmitters, we talk about them being excitatory or inhibitory, but really it's the channel that they bind to that determines if they're excitatory or inhibitory. So when we talk about our neurotransmitters, we'll talk about them being excitatory or inhibitory, but it's important to know that it's really the channel that determines whether it's excitatory or inhibitory.
Sorry, total question, just for curiosity's sake. The inhibitory, the chloride channel that the is going to bind to, is that like you're out exercising, your heart rate's up, and you're trying to get back down to resting heart rate? And like the chloride will come in and slow the heart rate down? Or is that?
So you've got the right idea. And again, this gets us very much into the autonomic nervous system, which we'll talk about next week. But here's the short answer. Absolutely. There is constant input to the heart to make it both beat faster and to make it beat slower. However, depending on what type of activity you're doing, the two inputs are not going to be the same. So yes, you get up and you start running around the room. As you get up and start running around the room, your sympathetic, that fight or flight, is going to increase. And as we release more and more of its neurotransmitter, which we'll talk about a little bit later, then it makes the heart beat faster. Once you stop running, right, then your sympathetic goes down and your parasympathetic can increase as well to help to bring it back down. Or if you start to meditate, instead of getting up and running around the room 15 times during our 15-minute break, you decide to close your eyes and get into the lotus position and start saying om. And as you do that, your rest and digest parasympathetic will increase, and that will release that acetylcholine and will slow the heart rate down. So yeah, this is part of that rest and digest process, this acetylcholine slowing the heart rate down. All right, so notice there are several ways. Remember, one of the key things we talked about, one of the key advantages to these chemical synapses are that they are modifiable. They're able to modify them. And there's several ways we can modify it. One, as we just mentioned, a very obvious way we can modify it is by having different types of channels. If we have sodium channels, it's going to be excitatory. If we have calcium channels, it's going to be inhibitory. But we can also change the number of channels. If our neuron releases 10 acetylcholine and our cell has one channel, is that acetylcholine going to have a very big effect? Let's say it this way.
More likely to bind.
Okay, possibly. But even if I released 100 or 1,000 acetylcholine, am I going to have a huge effect on this skeletal muscle cell if it has only one door that you can open to allow sodium in?
No.
No. right? Because you're restricted on how quickly that can get through. So you're not going to have that major of an effect. However, if we have 1,000 channels, then even releasing a small amount of acetylcholine is very likely to have an effect. And let's take the extreme situation. What happens if I remove all of the channels?
You're not going to get anything.
Yeah, you can dump a whole bucket of acetylcholine on there and it will be completely desensitized. It will have no effect. Conversely, if I add a new channel that has never been there before, I can now give it a completely different effect. So notice the types of channels and the number of channels are one of the ways that we can modify it. But as we also talked about, notice we can also modify it by the amount of neurotransmitter we release. We can do this a couple of ways. More or less action potentials. More action potentials will release more neurotransmitter, less action potentials will release less. We can also cause more vesicles to bind or less. If we release 3 vesicles worth of neurotransmitter versus 10 vesicles of neurotransmitter, we're going to get a different effect. And if you think about it, also how much neurotransmitter is in each vesicle. If we increase our production and we put 20 acetylcholine inside of a vesicle instead of 5, we're going to have a much bigger effect. So notice there are lots of ways we're able to modify this communication at a chemical synapse. Types of channels, amounts of neurotransmitters, the amounts of action potentials we produce. All of these are ways we can change the input, modify it. We don't want it hardwired. We want to have that ability to be able to change what's happening. Now, of course, the flip side of that is that when we have those type of chemical synapses, we know that also can be modified by medications and toxins. So we talked about botulinum toxin, or we talked about curare, or things along those lines, right? When you have this type of complicated communication system, it can be modified for good or for bad by other factors, toxins, right? Agonists and agonists, we'll talk about those a little bit later as well. All right, last thing we need to do is identify some of the common neurotransmitters. We already talked about acetylcholine. Again, I will say it, acetylcholine is the most common neurotransmitter in the peripheral nervous system. That, of course, means it's not the most common in the central nervous system. Those happen to be glutamate and GABA. Notice acetylcholine, as we talked about, because of different channels, can be either excitatory or inhibitory. However, in the central nervous system, glutamate is the most common excitatory, and GABA, notice GABA is gamma aminobutyric acid. And remember how I always say in this class, no abbreviations. I will actually allow you to use GABA. You do not need to worry about learning how to spell gamma aminobutyric acid. I don't care about that. But know that GABA is the most common inhibitory neurotransmitter in the central nervous system. Glutamate and GABA, even though they're so common, might not be ones that you're familiar with, but there are some others that you might be familiar with, like epinephrine and norepinephrine. And what's the other name that we use for epinephrine? Come on, I know you know.
Like the short version, like epi, like an EpiPen.
True, well, okay, but that's an abbreviation, but epinephrine actually has another name. No one says they're an epinephrine junkie.
Adrenaline.
There you go. Epinephrine and adrenaline are basically interchangeable terms. Epinephrine and adrenaline, same thing. And norepinephrine and noradrenaline. Again, either of those two names are acceptable. What do we use adrenaline and noradrenaline, or epinephrine and norepinephrine for?
Flight or fight.
Yeah, they're part of our stress responses. Mobilize our energy, mobilize our resources to help us to either fight that bear with an axe or outrun somebody in the room. We've talked about dopamine. We talked about the importance of dopamine and how dopamine production is lost in Parkinson's patients. But dopamine also is, we're finding more and more, plays a really important role in a lot of social interactions and social anxieties. Dopamine levels we're finding can influence and affect a lot of those types of factors, especially with all the high stresses of the pandemic and everything else that's going on. There's a lot of research and money going into dopamine. There's some larger, more complicated neurotransmitters like our friends endorphins. What type of things do you do that increase your endorphin production?
Workout.
Working out's a good example. What else? What else increases your endorphins?
Adult time.
Okay, yes. I was thinking of falling in love, absolutely. But yes, adult time can do that as well. And also, as it turns out, chocolate stimulates the endorphin. So you get very much the same effect from falling in love as you do from eating chocolate. Most of you haven't heard of endorphins. You may not have heard of substance P, but anyone know what substance P, especially with a name like substance P, might be involved with? Substance P is the primary neurotransmitter in our pain pathways. Our pain pathways are some of our least understood neural pathways, but obviously they're incredibly important, and so it's a big area of research. And then there's some unusual things that can be neurotransmitters, things like ATP, some gases like nitric oxide. Notice I said nitric, not nitrous oxide, right? Nitrous oxide is what you were huffing last Friday at that rave that you were at, or maybe last Wednesday when you were at your dentist appointment. But nitric oxide is an important vasodilator to control the diameter of your blood vessels to affect your blood flow. And stuff like that. Again, your book's got a nice table that does a good job of identifying some of the common neurotransmitters, where they're found and what they do. All right, questions on that? All right, that is everything I wanted to do from a lecture standpoint for today. So let's go ahead and end that. And let's go ahead and start this. Our goal now is going to be to play major catch up on our brain anatomy. Last time we left off talking about the major structures, and we'll emphasize some of those again at the beginning of this, and then we'll finally get to our areas or regions and their functions that you're going to be responsible for. But let's go ahead and take our 15-minute break. So we will restart at, looks like that'll be 9:25. And I will start the recording at that point. So again, if you don't have it already, get your handout, brain anatomy list to that central nervous system list, because that is what we'll be working on. All right, questions on that? All right, see you in 15 minutes. All righty, let's go ahead and get started. We've moved onto our lab stuff because we've got some catching up to do. And we left off last time talking about just anatomical structures. Things like the nooks and crannies, right? Again, this is a gyrus, this is a gyrus, this is a gyrus, this is a gyrus. All these things are gyri, right? This is a sulcus, this is a sulcus, this is a sulcus. Right? All those things are sulci. Those are pretty simple and straightforward structures, generic structures. Now, as we also talked about, there are some more specific structures, like for instance, the lateral sulcus or the central sulcus. And even some gyri, like the pre-central and post-central gyri that we have there. But again, these are all structures. These are structures that can be identified on a real brain. We also in the classroom have models of the brain that with some time and effort can be identified. However, as you know, time is one of those things we don't have a ton of on the exam. Now, the central sulcus is a very consistent structure, basically starts at the front of the, or the very top of the head, and then kind of migrates forward down towards the lateral sulcus that comes across. So again, with some time, these are structures that can be fairly readily identified. But it can be challenging when time is an issue. And so one of the advantages of having this PowerPoint presentation is that we can use illustrations like this. With the color coding, it makes it a little bit more easier to see. Now, again, all of these terms that we've talked about so far are just generic structures. However, within each of these lobes, are unique clusters of neurons that have a specific function. So for instance, across these three gyri, there are a cluster of neurons that have a very specific function. So this cluster of neurons found in this portion of the frontal lobe is what we call an area, or we also sometimes use the term region. So this here, an area or a region, is basically a functional group that have some specific function. And again, as we've talked about, location is a big deal because these neurons in this location have a different function from the neurons that may be located in this region, and have a different function from these neurons, and have a different function from these neurons. So location is one of those things that is really important in our nervous tissue. So within these lobes, we have areas, or what we're going to call regions. They have a specific name, and they also have a specific function. And again, the very tip of the occipital lobe is a pretty easy place to find. These three parallel gyri are things that can be pretty easy to find. But again, when we look at a real brain, it can be a little bit tricky. Just like looking at the models in the classroom are one of those things that can be a little tricky, especially the first couple of times you're dealing with them. So notice one of the things that they do on our models, that they do on our charts, is they put numbers on them. So that as you're looking at a muscle, you can see that that muscle is muscle 11, and you can look at the key, and lo and behold, muscle 11 is the biceps brachia. All right. Again, your bicep brachia doesn't come out with a big 11 printed on it. And different textbooks, different model makers, it may be number 11 in one model and number 75 on a different one. So the numbers themselves aren't inherently meaningful, but they're good to use to be able to identify. I make a point of emphasizing this because of this right here. these numbers that you see right here, where is it? Are what are known as Brodmann numbers. Corbinian Brodmann was a neuroanatomist in the end of the 18th and early 19th century. And basically, being a neuroanatomist in the late 1800s and early 1900s, obviously they didn't have like functional magnetic resonance imaging where they could put somebody in it and show them an image and see what parts of their brains lit up and were active. He basically decided to take it upon himself to compare a bunch of brains. So he got a bunch of cat brains. And he looked at the first one, and he looked at the second one, and he looked at the third one, and he looked at the 415th of them. And he's all, you know what? One of the things I noticed is that every single one of these brains, while there are some variations in them, all of them pretty much have this raised ridge right here. And because it was the first anatomical feature that he noticed on all of these brains, guess what he called it? Not a trick question. What do you think he called the first area of the brain that all these brains had in common?
Primary.
How about area 1? And guess what he found called the second structure that all the brains had in common?
Area 2.
There you go. And 3 and 4 and 17 and 45 and 22 and everything in between. Again, this was over 100 years ago that he did this, and we're talking about it now. When he was naming these, when he was putting these labels on them, did he know what these areas did, if they did anything at all?
No.
No, he was just looking at anatomical features. Period. And these are consistent anatomical features that were found across all cat brains. And then from cat brains, he went to monkey brains. And from monkey brains, he went to human brains. And he found these consistent features and he gave them these numbers. Apparently he had a lot of free time on his hand, but the fact that over 100 years later we're talking about this is because what is remarkable is that there is a tremendous similarity with some of these similarity between these anatomical features and, as it turns out, function. It's not identical. As we can see here, some of the regions or areas may be comprised of multiple Brodmann areas. Notice here we have a Brodmann, we have a functional area that really doesn't correspond to Brodmann's regions at all. But there's a tremendous amount of similarity. And so because of that, we're still talking about them today. Now, you are not going to be responsible for memorizing Brodmann's numbers, right? I'm not going to hold you responsible on the exam. You don't need to have memorized that area 17 is the primary visual cortex. However, just like a number on a model, if on the exam I say, what is model number 14? What is the muscle labeled 14? If I say that, you know exactly what I'm talking about. So if I say, what is area 17, and I'm pointing to it here on the picture, then that should help you to be able to know what it is that we're talking about. So these can be useful as we're discussing and going through them to be able to identify these things and make sure we are all on the same page when it comes to the talking. All right. Questions on that? Let's draw our brain. Again, it kind of has this mitten appearance to it. Maybe make it a little bit bigger. That's not bad. As we know, this is divided into 4.5 lobes. We know that the central sulcus, starting basically at the top of the head, actually, let's stick with black since we did it for this as well. We have that central sulcus. And the central sulcus divided what 2 lobes?
Frontal and parietal.
Yep. Excellent. We also had this deep invagination that forms the thumb. What was this deep invagination called?
Lateral sulcus.
Lateral sulcus, excellent. And it primarily divides what 2 lobes?
Temporal. Frontal.
Yep, temporal and frontal. And then our 4th lobe is the lobe to the back here. And what was that one again?
Occipital.
Occipital, excellent. And again, there are anatomical features, but they're deep, so we really don't see them in the anatomy the same way we see the central sulcus and the lateral sulcus. So what I want to do is talk about each of these lobes, and in each lobe, I want to identify some of the regions. Now, there is one more bit of anatomical structure we can see on this. Notice we don't see the longitudinal fissure, and that's okay for now. But we do know that there is this raised ridge that is in front of, and this raised ridge that is behind the central sulcus. So let's identify those 2 structures. What is this raised ridge in front of the central sulcus called?
Precentral.
Central gyrus.
Excellent, the precentral gyrus. And the one behind it is what?
Postcentral gyrus.
There you go. Perfect. So let's move this. Oh, I misspelled parietal. There. I still misspelled it. Sexy fingers this morning. They keep effing up. All right. Central sulcus there. Postcentral gyrus there. Make a little lead. There. Perfect. Excellent. All right. So for each of the 4 lobes that we've got here, we're going to identify some specific areas or regions we are responsible for. The first of those, conveniently enough, is an area known as the prefrontal lobe. The prefrontal lobe, or the prefrontal cortex, is the foremost, anteriormost part of the frontal lobe. And this is an area that is association cortex, associated with logic and reasoning and personality. Now, how do we know these things?
People that have had damage to it.
Exactly.
Yep.
One of the greatest discoveries in the history of the field of neuroscience is the development of helmets. because before helmets, young men went off to war and they died. After helmets, young men went out to war and they came back brain damaged, right? Damage to their brain, but at least still alive. And so a young man would come back and see the back of his brain had been blown off during war and he couldn't see anymore. And we'd be, whoa, lo and behold, the back of the brain must be where we use our vision. And this is also true for our prefrontal cortex. Plenty of examples of individuals who have been damaged in war and things along those lines. However, one of the most famous cases was an individual by the name of Phineas Gage. Anyone know who Phineas Gage is?
Yeah. Wasn't he the guy with the rod through his head?
Exactly, the guy with the rod, exactly. He was a foreman on the railroad, right? Back in ancient times, there were things called trains, and trains were the greatest modern invention at its time, revolutionized travel. And so building trains became a huge profitable career. And to be a foreman, you had to be really, really responsible, because one of the primary foreman's jobs was to handle the dynamite. Because if you're building your train and you come across a hill, are you going to waste the time and energy to build your railroad around that hill?
Negative.
No, you're going to blow that hill up so your train can continue to go straight. So what they would do is they would drill a hole into the rock, and then the foreman would put the dynamite in there and had this big long metal tamping rod where they would use a little hammer to tap the rod to get the dynamite down to the appropriate depth so that when it exploded, it would blow up the rock and they'd be able to remove it and continue forward. Where good old Phineas was tamping his dynamite down and the dynamite went off. And that rod shot out of the hole, hit him in the forehead, went through his forehead, out the top of his head, knocking him back 10 feet, and yet shockingly, he survived. Well, he lived anyway. However, his obviously tremendous brain damage to the front of his brain as a result of this, and one of the major implications was a complete change in his personality. Like I said, he went from a very even, mild-mannered, even-tempered individual to, if you read some of the reports, he became the kind of man who would drink during the day and curse at women, right? Totally changed his personality, right? Animal research would look at stuff like that, and they soon realized that your personality was located here in your prefrontal cortex, your reasoning, your intellect, your personality. And if we could modify it, we could modify your personality, right? This was a revolutionary finding at the time, right? Before then, if you had some type of mental disorder, oh, I don't have my microphone plugged in. Can you guys hear me okay?
Yeah. That way they lobotomize people through the nose.
That's exactly what we were going to talk about, right? If you had some type of mental disorder, right? If you were, for instance, a male who liked other males, or you were a female who was too often expressing your own opinions on things, right? These abnormal behaviors that nobody wanted, they would lock you up into a sanitarium. And was a sanitarium a place for people who are mentally disturbed to get better?
Nope.
No, basically their job was to lock you up so that you didn't hurt yourself or anybody else. But now suddenly they had a technique where they were able to modify your behavior. And you're absolutely correct. Basically, what they had was a metal tube. This metal tube was a big, long metal tube. Let's make that white. A big, long metal tube. And that metal tube would have a slot in the top of it. They would then put in a long piece of razor wire that was longer than the tube, so that when they pushed the razor wire up into the tube, that razor wire would bow out of that slot. And then you're absolutely correct. what they would do is they would take that rod, insert it into the nasal cavity, and break it through the cribriform plate. Once they broke it through the cribriform plate, they would then push the wire through so that the wire stuck out. They would rotate that wire around, basically cutting the axonal connections in our prefrontal cortex in, as you mentioned, a process called a frontal lobotomy. And of course, were you well anesthetized in a sterile environment while this was going on?
Probably not.
Absolutely not, right? Because then how would they know if it worked? Not surprisingly, many of the patients were quite apprehensive, dare I say, even nervous and concerned going into these processes. And basically, they would continue to destroy their prefrontal cortex until they weren't anxious anymore. There were doctors who specialized in this. One of them was a doctor by the name of Watson, who would tour the country. He had these cannulini, that's what these things were basically called. He had two of them that he kept in holsters, one on each hip. He would do both nostrils at the same time, and he would compete for speed. He would go to some local hospital and compete against the local doctors to see who could do them faster. Hopefully, you are appreciating how incredibly barbaric this behavior is. And it truly is. And there's no justifying what they did. But you have to at least appreciate and understand why they did it. Their goal was to try to make people better. Like we talked about before, a sanitarium was a place where you put people who basically had to stay there till they died so they didn't hurt themselves or anybody else. So the idea of being able to modify someone's behavior to allow them to go back and lead a meaningful life and, and to mesh with society was a valiant goal. They went about it in an incredibly disgraceful way. And more importantly, what was the other problem with this?
Infection.
Well, yeah, there wasn't just one, there was many. Infection was a major issue. Obviously, you're breaking a hole into the cranial cavity, so the leaking of cerebrospinal fluid could cause severe migraine-like symptoms and things along those lines. But the other problem is it didn't always work. Did some people get better? Yes. Did other people get worse or see no change? Yes, absolutely. And that's aside from the fact that it is a completely barbaric practice, which is, of course, why it ended. Why we realized that what we were doing was wrong, and so we stopped doing it. Is that what happened?
No, I just started doing it in secret.
Well, okay, I won't get into the conspiracies if it's in secret. I will say that no, it's not. It's in all likelihood, it is not happening anymore. Maybe there's some secret society of lobotomizers that is going on or something like that. But no, as general practice, it isn't done anymore. Not because it's been moved into secret and not because people thought better of it. But why don't we do lobotomies anymore?
Human rights.
You guys are so adorable and naive. Nope, we don't care about people. We found something cheaper and easier. Why
Good.
pay some schmuck like Watson who has to be a specialist to do this when we can, from a pharmaceutical company, buy a tiny little pill by the thousands? Yes, I have no idea what this pill is actually doing to an individual, but I know when I give it to person A, it changes their behavior into a method that is that I'm more comfortable with. I don't know why it changes his behavior. And while it changes person A's behavior, it doesn't change person B, and person C gets worse. But that's okay because I have dozens of other pills. So I'll randomize my combinations, trial and error a bunch of different ones till I find just the right a combination of chemicals that I have no idea what the long-term or even short-term implications of what they are, as long as they change that person's behavior so that kid can sit through an hour and 20-minute class and listen to lecture without bouncing up and down in their seat. What we are doing with medication for mental disorders is just as barbaric as what they were doing with the frontal lobotomies. It's just not as gruesome. Hopefully, 10, 20 years from now, we will look back at the medication of medical patients, mental patients, now in the same way we look back at these frontal lobotomies with horror. Oh my goodness, why did we ever do things like that? And hopefully we'll be doing something better at that time. But yeah, the reason we're not doing it anymore isn't because we, felt bad about what was happening. We found something cheaper and easier.
No, we'll just start seeing advertisements. If you know somebody or have a loved one who's taking these pills.
That is the cycle of life, isn't it? All righty. Questions on the prefrontal cortex. All right, next area. Let's go back to our drawing. I'll get off my soapbox now. As I mentioned, there are these three kind of parallel gyri that are located right next to each other in the frontal cortex. We see that here. And let's go back to the pretty picture here where we look at it here. Let's go back to this picture. So notice these two here, Brodmann's areas 44 and 45. And this is what is known as Broca's area. Of course, why is it called Broca's area?
Bob Broca.
Exactly.
Yeah.
Good old Bob Broca was the first person who identified it. Again, as we talked about, there are all these antiquated terms that are used, but one of the nice things is that we are getting a little better in some of these. And so this region now more appropriately is referred to as the speech area. And guess what the speech area is responsible for?
Speech.
Yeah, production of speech, right? Like what I am doing right now. Often we can get an appreciation for how these areas work, like we saw with Phineas Gage, by what happens when they are damaged, right? If someone had a damage to their speech area, they would not be able to produce speech with the same meaningful cadence that I am now. So instead of saying, Bill and I are going to go to the store to buy milk, they may say, Bill, store, milk, right? So damage to this area disrupts our ability to produce meaningful speech. We can still understand speech, right? If I tell that person to wave their left hand, of course I know that's my right hand, but you get the idea. They're able to follow directions. They can understand speech. They just can't produce it. Right? That inability is a condition we call Broca's aphasia. And aphasia is that inability to produce that speech like we talked about. And that Broca's aphasia is that inability to produce meaningful speech. Now, what's interesting about this area is for most individuals, it is located solely on the left side. Or in the left hemisphere, let's say it that way. So we're using more better terminology, more better. It's mostly on the left hemisphere. In, I would say, over 95% of right-handed people, it is found on the left side. And even like 75% of left-handed people have it here on the left side. Some have it on the right. Some have it in both. And again, how do we know that? Now it's easier because, again, we put you into one of those functional magnetic resonance imaging machines. They measure changes in essentially oxygen flow to different parts of the brain. So when you talk, one side of your brain will light up versus the other.
But we didn't have those machines 20 years ago. So instead, what they would do is they would inject an anesthetic into your carotid artery. And that anesthetic would basically, because you have the two carotid arteries, and for the most part, although there is some intermixing, your left carotid artery feeds the left side of your brain, and your right carotid artery feeds the right side of your brain. So they would inject the left carotid artery, anesthetize the left side of your brain, and then they'd ask you to say your name. And if you couldn't do it, it's because that's what side your Broca's area was on. Now, if it's just on one side of the brain, then what's happening on the other side? Anyone know?
Crickets.
Not quite crickets. Again, this is prime real estate, so we're not going to let it sit there and do nothing. It appears to play some role in mathematical, computational type of functions on the right side is what seems to happen. And here's the other thing. This speech area is highly specialized, right? You may not have thought it this way, but you're aware of that. Because if someone has a partial or some types of stuttering can be caused by partial damage or problems with the connections in this Broca's area. So if you have speech problems where you stutter, what's one of the ways that they teach them to try to get around it? Anyone know? A common technique that they give stutterers to help them to produce more smooth speech?
Isn't like we're like reciting like poems, like complex
Close.
or not complex.
Not so much poems, but you have kind of the right idea. They have them speak in a sing-song type of voice. I say that I am going to the store with Bob to get milk, and I will say it in kind of a sing-songy way, because singing is a very different function from speech. And you may not have thought of it in those terms, but I'm sure you're aware of it, because everyone in here has heard Adele sing one of her amazingly beautiful songs. But how many of you have actually heard Adele speak?
Does she have like a Cockney accent?
A hugely thick accent, right? Unless you're familiar with that accent, you pretty much can't make out a single word she's saying. She has a massively thick accent when she speaks. But is that accent there when she sings?
No.
No, because speech and singing are two completely different functions. This area is so incredibly specialized for speech that singing doesn't even pass through it. Even though you're literally singing words, singing is not speech. And so, and again, Adele's not the only example, but there's plenty of examples of singers who have very thick accents when they speak, but no accent at all when they sing.
Like the movie My Fair Lady. Catherine Hepburn.
Not familiar with that one. Does she sing without an accent?
Yeah, it's
Part of the plot.
Pretty good.
Cool, excellent. I'll have to check it out. All right, excellent. So that's area 2 in our frontal lobe. We have one more area, and this is where things get a little tricky, not because I think it's confusing, but again, as we've talked about, we have to be careful with our vocabulary. The third region of our frontal lobe we have to know, and let's start easy. The area is our primary motor cortex. What do you think our primary motor cortex does?
In charge of movement,
Of what?
muscles,
Which ones?
our primary,
Close.
our skeletal muscles.
Which ones? I know you know the answer. Take it the last step. My primary motor cortex over here on my left hemisphere controls which
controls your-
skeletal muscles?
On the right side of your body.
Right. And what's the correct term we use for when it crosses the midline like that?
Contralateral.
There you go. So our primary motor cortex is the region of our cerebral cortex that is responsible for voluntary control of our contralateral skeletal muscle. So when I want to wave my right hand, I make that decision over here in my left primary motor cortex. I think hopefully that's pretty simple and straightforward. Yes, we comfortable with that?
Yep.
Here's the tricky part, where it's located. Our primary motor cortex is located on the precentral gyrus. So notice on the exam, if I have an arrow and I'm pointing to this, if I have this arrow right here, if you notice, there are really 3 questions I can ask. I can ask the ones we'd expect: identify the area, identify the function of the area. But I could also ask you to identify the structure. All right, so let's do that. What is the area that this arrow is pointing at?
Central gyrus.
Nope. Remember, the
primary
area is
motor.
functional. There you go. The area, and actually it'd probably say area/region. That is the functional name. So that is the primary motor cortex. If I asked you for the function of the area, what would your answer to that be?
In charge of voluntary movement of contralateral skeletal muscles.
Perfect. But if I instead asked you to identify the structure, what structure am I pointing at?
Precentral gyrus.
Precentral gyrus. There you go. So in this case, the area just happens to be on a gyrus that has a name. Now, I'll tell you a secret. All these gyruses have names. The 3 gyruses that make up Broca's areas, they have names as well. However, do you care?
No.
No, because they're not on your exam, right? But in this case, this one gyrus does have a name. And it just happens to be on that specific named gyrus, happens to be the area that we are interested in. Okay, I'm going to cheat and move this over here because I need it out of my way. So, all right, if I'm going to put this up here, move that there, move that there, and then I can bring that there. Perfect. And now my parietal region's open. All right, excellent. So notice if we go back to here, this precentral gyrus is where our primary motor cortex is located. Now, one of the cool things about this region, here we see it. Notice this picture, the brain goes the opposite direction. There's our primary motor cortex on the precentral gyrus, but this is what I wanted. Here is that precentral gyrus. This is our primary motor cortex. And what we actually see is that there is actually a map of the body along the surface of this cortex, so that different regions control different parts of our body. And again, you may not have thought of it in these terms before, but if you have an uncle who maybe had a stroke is often caused by a blocking of a blood vessel. And when that blood vessel, there's a big, large blood vessel that goes down our central canal, stopping blood flow to some part of the body. And suddenly now he can't move his hand and his wrist or his arm. Face is fine, trunk is fine, knees, legs, toes are fine. All of those things are fine. But the small part that has been damaged is where he's lost the ability to have that movement. Notice also that the size of the cortex used for processing information isn't necessarily equal to the size of the part of the body. Notice the largest representation is the face, especially the lips, the hands, especially the thumb, right? And again, if you think about all the muscles here that allow us to get all of that sense from your loved ones when they're confused or they're happy or they're aroused or angry, they can get all of that from their facial expressions, right? How many of your loved ones have expressive knees where you can tell just by the muscle contractions around their patella whether they're angry or upset or sad or happy? Right? We don't have that kind of muscle control. We don't have that kind of expression. And so not surprisingly, we don't have nearly that much region of the brain associated with that. And notice one other thing that this picture shows us as well. Right across the central canal from the primary motor cortex on our post-central gyrus is our primary somatosensory. Our primary somatosensory cortex basically provides us with our sense of touch from the contralateral side of the body. And the maps line up pretty well. Notice the part of the brain that moves the hand is right across from the part of the brain that senses touch from your hand. Which is also why when your uncle had that stroke, not only did he lose movement of his arm, but he also lost sensation from it as well. Now, is it a perfect mirror? No. He may have lost movement from the elbow down, but only touch on the hand. He can still feel if you touch his forearm. And again, as we talked about, notice the areas of the cortex that have the greatest representation, the hand, especially the thumb, the face, especially the lips. They've taken these representations and actually used them to make these great models. I couldn't find a really good picture, but here's one that I have where you see this structure that is known as a homunculus. The homunculus is basically an illustration of a human based on the amount of cortex that is representing the different regions. So notice over here, we have our motor control, huge, massive control of the hands, especially the thumb, huge control of the face, especially the lips and tongue, very little for the rest of the body. And the same thing for our sensory as well. Notice here we see ears because we can hear. But again, most large representation of the face, especially the mouth and tongue, huge representation of the hands, and less to the rest of the body. So these funny looking creatures are what are known as the homunculi. And they again give us that representation of the amount of our cortex on our primary motor cortex, and now also our primary somatosensory cortex. And notice with our primary somatosensory cortex, we can ask those same three questions. I could ask you to identify the region or area. I could ask you to identify the function of the region / area. And I could also ask you to identify the structure. Notice That's not the same for this here or for this here. For these two, I really can only ask 2 questions. Identify the region or area and identify the function of the area or region. So because the primary somatosensory cortex sits on the postcentral gyrus and the primary motor cortex sits on the precentral gyrus, there's a third question I could technically ask with those arrows. Whereas these black arrows over here, there's really only 2 questions. All right.
Could you put the for the the primary somatosensory cortex? Can you put that on this diagram as well, by chance?
Yeah, that's the blue part here. This is the primary.
I mean, like the written part that you had
okay.
on that other slide.
Got it. Excellent. Which, as we now know, is contralateral touch. All right. Questions on that? All right, there is a second, I think only, yeah, a second region of our parietal or area of our parietal lobe we're responsible for. This one's shape is a little more ambiguous. It kind of is very close and sometimes actually wraps around the lateral sulcus. Let's look at some of the representations they've given for us in the, here we go. So here on this illustration, it's these areas, 39, 40, and a little bit of 22 like that. Notice here, they've colored it in for us. And I know different models have the brain pointed in different directions, so don't get confused by that. There's the lateral sulcus, and here's that area here. And we see how it compares to our speech area, our Broca's area here. This general interpretive area is also what is known as Wernicke's area. Of course, why is it Wernicke's area?
Oh, Bob Warnicke.
Good old Bob Wernicke was the first one who identified this. Again, general interpretive gives you a better sense of what it does, though. Remember, Broca's area is where we produce speech, but our general interpretive is where we make sense and produce meaningful. Speech. Once again, someone who has a damage to this area has what is known as Wernicke's aphasia. And instead of describing it to you, this time I'm going to show it to you.
What's this?
Can you guys hear that okay?
Yeah.
Okay.
Yes.
You know, the big right there, end of the football.
End of the football?
It influence the football right here. The cause that it comes in, see?
Yeah.
The end of it, see? Yeah. Oh, we have out of it, see?
I don't understand.
Well, it's pliable in the other, see? You don't understand it, but it plies in the long tree comes from there. And it comes source from the country out through, see? All the under it, see, it comes up and finally come out here and goes out, see? Get it?
Sort of.
Yeah, it comes out here. See, it comes out here, here, you and here,
Uh-huh.
and it comes in,
Uh-huh.
and it comes out, and bing. That's a good fly throw. See,
That's a good what?
that's a good flow. Flow goes fly. See, it throws cold, or it gives, or kicks out, or bing, or whatever it is. We has no football about it out. It's just pulling. See, but they say feel it. Oh, I guess you meant you have beds. See, I don't know.
Where do you live?
Where do you live? You're in your live. You're up here. See,
Where do you live, Doc?
where do you live?
Where do you live?
Where do you live? Is well, if there's a run through here around here. You?
You used to be a dentist.
Yes,
Yeah. Where?
right here. the rest of the period. Yes.
Yeah. How many years were you a dentist?
How many? Holy Christmas. Oh, about 100 and, let's see, 130, about 40. No, gosh, what number is it? One plus 5. Wait a minute, no. It's a pretty close to get it this one day. It's fine over there loft, see. It's claws. Oh, it goes for about over 30, see, or 130 for down, see, in front of out for it goes against her, and then it comes back to the against her for 4, and then goes out for the rest of the time. The third is not the servant, but the soldiers, where they have the soldiers of the suburbs and the suburbs and all the first suburbs, and watch out in the short side, and they cross with the sugar 2 hours, 2 high or low. Wowie, God, these are terrible. Wow, these are terrible words.
These are terrible words.
They sure are.
Hard?
Yes.
What you're saying, you mean?
Yeah, that's what they do. They actually go them a lot harder. It's when they harder in hard go, and then when they go out, finally they quit. See, the heck of a time don't do it.
So notice if you were sitting at Starbucks and this was going on the table behind you and you really weren't paying attention, it sounds like they're having a conversation, right? His speech has the cadence, has the speed, it has similarity to speech. But it isn't until you actually stop and listen to what he's saying that you can see that a lot of it isn't meaningful. I saw someone right during the chat. Oh, my chat window went down because I'm not sharing anymore. Here he is. Yeah, Emily's got a patient like this, and it can be very challenging. And it's also very difficult when working with these patients to know what it is that they understand and what they don't. Notice you can clearly, and again, I've watched this many times, so some of these more subtle things you may not have picked up on, but let's talk about some of them. One of the things he has difficulty is understanding speech. So for instance, she asks him, where do you live? All right. Now, if I were to ask that to you, where do you live? And you were sitting there, you might want to think about that for a minute. Do I really want Dr. Slutsky to know where I live? And while you were thinking about that, you would switch the question around. Brian, ask me.
Where do you live?
Where do I live? Right? I turn the question around because I'm thinking about what the answer is, what I want to give you is. But she says to him, where do you live? And he repeats it back verbatim. Where do you live? Where do you leave? Where do you live? He's not understanding. He's not comprehending the words. So clearly there's some things that he can't understand. Samuel, great questions. Depending on the damage, some people can read and write without any problems at all, but others will have problems with written text, both writing it and reading it. Obviously, other questions he definitely understood. You were a dentist, where? And he points at his teeth and laughs. Then she asks him how long. And he spends like 5 minutes, it seems like, trying to figure that out. Now, it sounds like, because again, I've watched this many times, what his challenge was is that it looks like he became a dentist and then had to go away to war, probably World War II or something like that, because he's talking about, you know, going around the backside and flanking and the soldiers and all that kind of stuff, and then came back from the war and was a doctor through the rest of the time that way. So it sounds like that's what he was trying to explain. And then again, it's hard to understand how much of a deficit they're aware of. But even at the end, he does show some frustration, right? He shows that these are hard words. And then he talks about how he seems to be okay when, you know, it's fine working around in here, but as soon as the words come out, they just die. Right. So clearly he has some frustration, but how much they understand their deficit, how much they're able to comprehend, these are really challenging aspects.
It's almost like he knows what he's saying and what he's trying to say, but it just comes out as garbled, and it's very, I can imagine that'd be extremely frustrating.
True. And so based on that, the fact that he laughs, the fact that he has a pretty good demeanor, you got to wonder, is he really gotten to the point where he's tolerant of this, or how much of a deficit does he actually understand that he has? And that's part of the challenge, is we can't do that. Emily, you were about to say something?
I was just going to say that's exactly it. I mean, with my patient, you know, she definitely knows what she's trying to say, and then it comes out and she does the same thing where she gets frustrated, she'll laugh at herself. And it's really hard when you're caring for someone like that, because they can't really communicate what they need. And over time, you kind of start to understand their language and the way that they communicate certain things. And there are certain like repetitive things that they say that, you know, it means a certain thing.
Yeah.
But it's really hard.
Yeah. And again, like I said, I've watched that video many times, so it's allowed me to pick up on many of the subtleties of it. But yeah, it's very challenging. And like, in many ways, like dementia, like Alzheimer's, like things like that, in many ways, it's harder on the family and harder on the caretakers than it is on the people themselves, because it's hard to tell how much they're fully aware. Clearly, they are aware there is some deficit and they're having problems, but how fully aware they are of it isn't a question you can easily ask them. The last thing I want to point out about this that is incredibly interesting is this is another one of those characteristics that has, and again, we mentioned it earlier, but I don't think I used the fancy vocabulary term for this. Both of these areas have what we call lateralization. By lateralization, what I means is, unlike touch, unlike movement, which is on both sides, speech and general interpretive, here, hold on, I didn't write that. These areas are lateralized, almost completely found on the left-hand side. Like I said, almost 100% of right-handed people have it over here on the left, and even something like 80% of left-handed people have it over here. About 10% have it on the right, and about 10% have it bilateral. So what's on the other side? Well, it turns out for the general interpretive answer, on the right side is actually spatial awareness. This is another one of those characteristics that is highly, highly specialized. Again, like Emily's patient, it is interesting to talk about these things in just the clinical sense, but when you're actually dealing with the people, it is much, much more frustrating. This is another one of those that's really interesting. If you had a patient who had damage to the right side of basically what would be Wernicke's area, and you asked them to draw a picture, you brought out a potted flower and asked them to draw it, basically what they would draw would be this. basically they ignore everything to the left. Their spatial awareness of the contralateral world is unaware. They only draw the right side of things. They will only dress the right side of their body. They will constantly get angry of whoever this strange dude is that's so close to them that their arm is right next to them in their personal space. They won't wash the left side of their body when they shower. They won't dress the right side of their body. I had a student, gosh, 5-6 years ago now, whose boyfriend was in a car accident and damaged that spatial awareness on the right side. And she, you know, it wasn't a complete damage. So again, but there were times where he would forget to wash the left side of his body or would forget to dress the right side of his body. I mean, the left side of his body, things like that, because you lose that awareness of that contralateral side of the world. All right, questions on that? All right, we are halfway through our lobes and rapidly going through the time, but again, I want to keep us fresh for this. So let's go ahead and take our last break. We'll take one last break, come back, get through the rest of the lobes, and then see how far we get through the other cephalons. But let's go ahead and take a break first. Let's come back at, looks like 10:43. So one last break, and I will start the recording at that point. All right, any questions before we take our last break? All right, see you guys in 15 minutes. All righty, let's dive back in. Any questions before we get started? All righty. From the parietal lobe, I want to move on to the occipital lobe. And in the occipital lobe, there are kind of two main types of areas that we are interested in. The first of which is the very back of the occipital lobe, the very back of the brain. And this is a region that is known as the primary. There we go. Visual cortex. Now, just like we did for the primary motor and the primary somatosensory, we identified it. So what do you think the function of the primary visual cortex is?
In charge of processing visual stimulation.
True, but again, we want to be more precise. This is the first location where we receive information.
It's not typing.
Oh, that's not writing. Hold on. I wrote that on the bottom of the screen. Hold on. Let me cheat and grab that. Didn't work. All right. So this is the first location where we receive visual information about what?
World around us, what we observe.
True, but notice over here on the left side of my brain, do I receive sensory information from the entire surface of the body?
On the right side.
True.
From the right eyeball.
Contralateral. And in this case, and this is where vision gets a little bit interesting, we receive information about the contralateral side of the world. Notice I don't mean the right eye goes to the left side of the brain, because here's what I want you to do. I want you to cover your left eye. If you cover your left eye, do you just see information from the right side of the visual world?
No, there's overlap.
No, there is some overlap between the two. So it gets a little bit trickier. The way this works, and here's a great illustration from your textbook that shows this,
is notice, for instance, let's stick with the right eye we were looking at. This is a top-down view of the brain. And we see here from the right eye, we get information both from the right side of the world represented in blue and the left side of the world represented in red. The stuff that we are getting from the right-hand side of the world, we want to go over to the left hemisphere. So notice that information here in blue, and actually I should probably change the size of this. This information here in blue, notice has to cross to get over to the left side of the brain. Whereas, and I'll change colors, the stuff from the left side of the world coming to the right eye is already on the correct side of the body. So we want it to stay and go here to the right side. And the same thing happens with our left eye. Our left eye gets some stuff from the right side of the world, and that stuff we want to stay over here. So this information stays over on this side. Whereas information from the left side of the world needs to cross to go over to the right hemisphere. So here in the center, we have this region where we have a partial crossing of information. These structures, of course, have names, and we'll identify those when we get to the diencephalon that they are a part of. But the important thing to remember is we have this special structure known as the optic chiasm that allows for information to cross. Notice up here, we are going from eye-specific information to, in the brain, we go to side-specific information. So everything that is received to the left side of the brain comes from the right side of the world. Everything received to the right side of the brain comes from the left side of the world. So we have that nice crossing of information. Now, you may not have ever thought of the primary visual cortex before, but you may be aware of it. As we talked about, which you should not do, if you were to put pressure on your eyeball, you would increase the interocular pressure, and you would see lights as a result of it. Well, if instead someone punched you in the head, and as they punched you in your head, your brain was vibrating around inside of your skull, the back of your brain, that primary visual cortex, could get mashed across against the occipital bone. And when you put that pressure on the primary visual cortex with the occipital bone, what happens?
Go blind for a second.
Well, true, you either could go blind for a second, or what else could happen?
Is it a concussion?
It could cause a concussion, although remember, concussion is more affecting your reason, your intellect, your personality. So that's more when your prefrontal cortex gets smashed up, because as you come back, it bounces back, right? Think of the cartoons. When someone gets hit in the head, what do?
Blackout.
They don't black out in the cartoons. What happens to them in the cartoons?
You see little birds.
They see little birds, or they see stars, right? Those lights that they see are that pressure on the visual cortex. Our primary visual cortex, as I mentioned, is where we first receive the information about light. And basically, it is the most basic information, right? Light or no light. Again, let's think of this in terms of damage. If your primary visual cortex was damaged, what would happen to you? What do you think?
Receive light.
Yeah, you'd basically be blind. You wouldn't be able to see any light. Nothing. You would not be able to see any light at all. Now, obviously, getting light is important, but we also have to process that visual information and make sense of it. And to do that, oh, I'm not sure how I did that. Oh, that's how I did that. There's a bold. How clever. We have our association visual cortex. Why is this not writing? Our visual association cortex is where we process the light information and make sense of it. Some of this is very basic. Right next to area 17 is a region that is Brodmann's area 18. And area 18 is the part of the brain where we perceive and make sense of color. So notice if we damaged your area 18, you would be able to see light, but you wouldn't be able to see color. And it gets far more complicated than that. If you remember, as I mentioned, over 40% of our brain is involved in processing visual information. And as it turns out, we have two big pathways, one that even leads into the parietal area, one that even leads into the temporal area. And these two pathways are essentially what are known as the where pathway and the what pathway. Guess what the where pathway is involved in processing?
Where?
Yeah, where we're perceiving a light. It doesn't matter what we're seeing, whether it's a blinking red light or whether it's your grandmother on a skateboard. These cells don't care about that. They care about where they are and where they're going, how they're moving. Again, one of the ways we can understand and appreciate how these systems work is when they're damaged. Up here in the where pathway, I'm going to write this down, but I do not want you to memorize this. There is an area known as MST. And MST is the area that plays a role in our understanding of optic flow. Anyone understand what I mean when I say optic flow? Apparently not. Optic flow is how the world moves around us. Notice if you were sitting in my chair and I spun you around, you would see the world moving by, right? Or if you were to get up and walk forward, notice as you walk forward, the world around you comes up and kind of expands around you as you're moving. That movement of the world as you move through space is what is known as optic flow. And people who have damage to MST are not able to perceive optic flow. Well, that sounds crazy. What might that look like? Well, as we said, back on Friday night when you were sucking down that nitrous oxide at that rave, one of the things they will frequently have at a rave is a strobe light. Anybody ever been in a room with a strobe light on before? I'm not going to accuse anybody of being at a rave, but have you ever been in a room with a strobe light before?
So fun.
Excellent. If someone's waving to you in a room where there's a strobe light, do you see the continuous movement of their hand?
No.
No, their hand is here, and then suddenly 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 their hand is there. That is what life is like if you have damage to MST. You are constantly living in a strobe light.
It's like a horror film in life.
Exactly. Can you drive a car if you have damage to MST?
No.
Not very well.
Hopefully not. You won't. Can you pour a cup of coffee? Can you pour a cup of coffee if you have damaged MST?
No.
No, because here, and then suddenly it's here, and then suddenly it's overflowing. Except here's the cool thing. People are really clever. People with damage to MST do pour themselves cups of coffee. How do they do it?
Stop by sense of touch.
Yeah, exactly. They put their thumb in the mug, and you basically pour till you feel it touching your thumb, and then you know to stop. But yeah, imagine life living in a constant strobe light. That is what MST is. What pathway doesn't care where the object is in the world? It's all about identification. Again, this is not an area you're going to be responsible for, but I want to mention it because it emphasizes the point. There is a region of cortex in the what pathway known as the IT region, intratemporal. And it is one of the major areas that is responsible for identification. There is a classic case of an individual, a famous individual who has damage to their IT region, and a tremendous amount of research has been done in him. And one of the most famous cases is he was shown a glove. They showed him a glove, and it was yellow and blue stripes on the glove. And he was able to describe it. He could see the colors. He could see that it had a central body with these 5 extensions coming off of it. He could clearly see it, and he could describe it. But then they asked him what it was, and he had no idea what it was. And so he sat there and thought about it for a while and thought about it for a while, and he saw those extensions coming off the central place. He said, oh, it must be a change purse. Each one of those extensions, like one's for nickels and one's for dimes and one's for quarters. They then hand it to him, and he can hold it in his hand and add that tactile sensation to the visual. And suddenly he realized it was a glove. And it doesn't just work for objects. This individual happens to be married. However, if his wife is standing still in the room, he will not recognize her. He sometimes can recognize her when she's moving. As she walks, he can sometimes recognize her by her walk. But if she's just standing there in the room, he does not recognize her as a person, as an object. He can see her, describe her, but doesn't know what she is. As soon as she talks, he instantly knows who she is. Like I said, very famous case. A lot of research has been done. A lot of books have been written. There's a very famous book written about him, The Man Who Mistook His Wife for a Hat. So there's a lot of really interesting research and information that has been done from this. But again, it gives you these 2 regions. While they're not things that I want you to necessarily memorize, I want you to appreciate what's going on. What is going on here is that we have this tremendous ability to make sense of that basic light information that is coming in. We have all this different light at different intensities and different wavelengths, and we take all that information and process it into an understanding of the meaningful world around us. All right?
So would damage to the where pathway affect depth perception?
Yes. I laugh at that because absolutely. Depth perception is a 2-fold process. One part of the depth perception occurs here at the levels of the eyes, right? Again, if you close one eye and close the other, you'll notice the world shifts around you, especially if you put a finger in front of you. If you put a finger in front of your face and look at your computer screen and you go back and forth between your 2 eyes, what you'll notice is your computer screen doesn't change that much, but your finger in relation to it does. So some depth perception is what we call parallax. Your 2 eyes are slightly offset from each other. And so as a result of that, you're getting 2 slightly different images. And those 2 slightly different images is what gives you your perception of depth. Again, and we've known this for a while. If you think back to the original 3D movies, basically they used 2 cameras taking the picture of the same thing. And one of them they tinted red and one of them they tinted blue. So what did you do? You wore blue and red glasses. One side was red, one side was blue.
The red on one side masked the red image, so you just saw the blue one. The blue one masked the blue image, so you just saw the red that were slightly offset. And those two slightly off images would make the movie seem 3D. Right? We do something similar with polarized lenses now, limiting what gets to the eyeballs. So yes, some of it is retinal, but that normal, it's about arm's length. So again, it's much easier to thread a needle with both eyes open than to do it with one eye closed. With one eye closed, that depth perception is a little bit trickier. But if you're landing an airplane, for instance, when you're landing an airplane, you're relying on cortical processes and the understanding. That's why those simulators are so effective. Because even though it's really just a two-dimensional screen you're looking at, you get that sense of depth. And again, this would be a perfect example where we'd be in the classroom. If we were in the classroom right now, I'd be standing behind the desk and you would not be able to see my legs. Would you think that I did not have legs?
No.
What do you think? Do you think I have legs? Yes, I have legs, right? But you wouldn't be able to see them. And because you understand how the world works, you would know that because you can't see my legs, that means the desk is closer to you than I am. And that gives you a sense of depth. So that type of understanding how the world works, that is what goes on in the brain. So yes, that type of information. Some of it is visual, right at the eyes, the optics of it. But yes, some of it is cognitive. And so some damage to the wear pathway could affect depth perception, yes. All right. And it doesn't just work for vision. Here in the temporal lobe, we have the exact same thing. We have a primary auditory cortex, which pretty much sits, it's a little, so I'll cheat. It's a little offset. The central sulcus kind of points at it a little bit, but it's a little offset. This is our primary auditory cortex. And this primary auditory cortex is where we receive the basic information of sound. Then surrounding it, oops, hold on, need that to stay orange, is our auditory association cortex. Which is where we make sense of that auditory information, right? Your primary auditory cortex is where you're hearing two tones. Your association cortex is where you realize those two sounds are oscillating back and forth. They're increasing in amplitude. And oh, that means that there is an ambulance driving towards me. I should step out of the street. So for both of these, there's an area where we get the general information, and then there's an area where we process it. Primary and association cortex. All right, and again, notice I've done an amazing job of drawing this, but we have all these pretty pictures from your textbook showing these things too. So notice again, here is our primary auditory cortex, and then the association is surrounding it. Primary visual, our association visual. And there are those. All right. Questions on that?
Hey, could you go back to the whiteboard for just 1 second, please?
Nope. And I'll save that. I can go ahead and save this and post it too.
Good.
Yep. All right. Any questions on these 4 lobes? All right. If you've noticed, and hopefully you have, because I haven't been super subtle about it, I keep saying
4.5.
I keep saying 4.5 lobes. The reason for that is because if you were to grab the lateral sulcus and peel it open, there is a big fold of the cerebral cortex found on the inside.
Notice this is a good view of it here, but actually one of the easiest ways to see it is with a coronal section. Notice with our coronal section, what we see here is this is the parietal lobe coming down. This is the temporal lobe coming up. This is the lateral sulcus. And notice on the inside here is a big, huge fold of gray matter. This big, huge fold of gray matter is what is known as the insula. And there are some anatomists who refer to the insula as a fifth lobe. Now, I don't care if you describe it as a fifth lobe or not, just know that this is the insula, but we need to know what the insula does. The insula is the cerebral cortex that basically coordinates and manages the nuclei that are deep in the telencephalon, or let's stick with cerebrum. Remember these nuclei in here, these multicolored things, all these fun things in here are clusters of gray matter known as nuclei. And collectively, they form a functional group that we call the limbic system. So our insula basically is the cortex associated with the limbic system. Now, what's really interesting about the insula is an area of the brain that is definitely associated very much with things like desire, things like anger, and probably most important, addiction. Oops, hope I spell it right. Addiction. Again, one of the things we're able to do with these functional magnetic resonance imaging machines is put a person in there and watch blood flow change in the brain while they do activities. That's what a functional magnetic resonance imaging or a functional MRI does. Because the implication of that is the more blood going to a part of the brain, the more active that part of the brain is likely to be. Needs more oxygen, needs more resources, it's more active. And that's how we've been able to observe the activity of this insula. What they've been able to do is take individuals and put it in there. And for instance, they could take a typical individual, put him in there, and then have them watch a coin flip and try to guess whether it's heads or tails. And they'll have them do that activity and keep track of how well they do. And then they take someone who's addicted to gambling and they put them in there and they do the exact same thing. And what they see is that person who is a gambling addict sees dramatically more neural activation in the insula than just a normal individual. They're getting this excess stimulation of this region, and they believe that this excess stimulation of this region may be one of the things that causes us to want to participate in that activity again. Think of the implications of this, not just for gambling, but for drugs, sex, all sorts of other activities. If we could inhibit the activity in the insula so that you didn't get that same reward from doing that behavior that you're addicted to, it might be much, much easier to stop smoking or to stop gambling or to stop doing drugs or things along those lines if we were able to inhibit that function. So again, there's a lot of research going into this insula. In fact, this whole limbic system is really, really important. Again, this whole area collectively is known as the limbic system. We see it nicely with this cross section, but your textbook's got this cool picture where we kind of see, here's a three-dimensional view. Here we have a partial sagittal section
where basically they've just removed part of one of the hemispheres, but left the temporal lobe intact. And here's another three-dimensional view. And we're able to see these nuclei that make up the limbic system. Now, I wrote this down because I want to read it verbatim, because I want you to understand this. I like this definition and I want to read it to you. Again, you don't have to memorize this definition, but it's useful and we'll talk about it. The limbic system is the link between our cerebrum and the diencephalon. It is a functional group involving emotions, memory, storage, memory storage and retrieval. And here's the part I wrote down because I thought that I liked the wording of this. It links conscious thought and unconscious emotions. So again, functional group involved in emotions, memory storage and retrieval, and it links conscious thought and unconscious function. What do I mean by that? Linking conscious thought and unconscious function. I did say these things out loud, right?
Yeah.
So what does that mean to link conscious thought and unconscious function? All right, let me ask the question this way. Here it is, Tuesday night. So this is the perfect opportunity because you have all day tomorrow off to be able to study. That you would want to go down to the local, I don't know, pool bar and shoot pool all night long and drink very heavily, pretending that you're studying. So you come stumbling home at 3 o'clock Wednesday morning when most people are sound asleep, needing to get up early to go to work or take care of the kids or send them to school and all that. You're reeking of alcohol, fumbling at the front door, dropping your keys, rattling the doorknob as you're trying to get in, making so much ruckus that your significant other comes down to unlock the door for you. And without even saying a word, just by looking at them, can you tell exactly how happy they are to see you? And that they're likely probably aroused and want to make sweet, sweet love to you as a result of your state? Can you tell that just by looking at them?
Oh, yeah.
Yeah, exactly, right? That whether it's that vein that throbs on their forehead, right? Or the flush to their face, right? Or the tightness in their jaw. Are they consciously thinking, I'm very angry at them, I should probably increase my heart rate?
No, it just happens.
No, exactly. What they are consciously thinking affects their unconscious body function. It's the whole point of a lie detector test, right? I hook you up to a machine and I ask you a bunch of benign questions. What is your name? What is your favorite color? Right? All of this kind of things along those lines to get your baseline of your heart rate, your blood pressure, the conductivity of your skin by how much sweat you're producing, how much you move around in your chair, right? Are you a cat person or a dog person, right? Do you hate Mondays as well? And then it's, where are you on the 5th, right? They want to know these questions because when you are thinking about lying or actively lying, does that have the ability to physiologically change the conditions of your body?
Yeah.
Yep.
Yeah, if you're wired that way. It's one of the reasons why lie detector tests aren't submissible as evidence. Because if you're a true sociopath, then you don't think what you're doing is wrong. And if you don't think what you're doing is wrong, is your heart rate going to go up? Are you going to increase the amount of sweat that you're doing or anything along those lines?
No.
No. Or, when they first started using a lie detector test, one of the things they would do is people would put a tack inside of their shoe so that when you answered a benign question, you'd push your toe into the tack so that you'd feel the pain and it would change the physiology of your body. And then when they asked the serious questions, you'd take your foot off of the tack and, right, it would level things out. So yeah, these things can be tricked, but the point of them is legitimate. The conscious thoughts that you have in your body, scared, sad, aroused, angry, all those emotions, all those things you think about have an ability to physiologically affect your body. And it is here in the limbic system where we link those two things together, what you're thinking and what's going on in your body. Let's talk about some examples. This picture shows it nicely here, although I like this picture a little bit better. Down here, deep in the thumb of the temporal lobe, is an almond-shaped nucleus, which is where it gets its name. Amygdala, or amygdaloid, means almond-shaped, known as the amygdala, or the amygdaloid body. The amygdaloid body is primarily responsible for helping us to process and recognize fear and anger. Humans are incredibly social organisms, and as we finished talking about, we communicate very well non-verbally. But we're not the only ones. Monkeys in particular are very social, work in clans, and communicate non-verbally. And with monkeys, there are two primary types of facial expressions. They have the aggressive, angry facial expressions, and they have the submissive facial expressions. And monkeys are incredibly good at telling them apart. They will train a monkey to sit in a chair, and they will show him a picture of one of these two faces, and they will know to push one of two buttons. If it's an angry face, push the angry button. If it's a submissive face, push the submissive button. And when they do the activity, whether they're right or wrong, they get rewarded with juice. They get a little sip of juice. And so they will sit there for hours and do this activity because they love juice. And they're really, really good at it. Unless they have damage to their amygdala. If they have damage to the amygdala, then they're no longer able to differentiate the aggressive from the submissive faces. In our house, we have a different name for the amygdala. The amygdala in our house is our worry monster, right? Your worry monster's job, recognize fear, recognize aggression. Its job's to keep you alive. Because back in ancient times, bears with axes roamed the countryside. So as you were sleeping in your tree outside or under the tree outside, if a branch snapped around the corner, it could easily be a bear with an axe getting ready to come and gobble you up. All right. Luckily, we don't have to live in trees anymore. Luckily, there are much, much fewer bears with axes roaming the countryside. But especially with that little kid in their bed, when they hear the creaking noise outside or the creaking of the house, right? Does their worry monster exaggerate that for them and make them believe that bears with axes are coming to eat them up?
Yeah.
Yeah, absolutely. Right. So that's that worry monster, right? You got to get out of your amygdala and get back into your frontal lobe. Right. So there are all sorts of techniques that you can use to get out of that worry monster, get away from that. Again, very, very healthy from an evolutionary standpoint. But when it's 3 a.m, right, and you're still worried about those boogeymen, then it becomes a lot more challenging. So that's that amygdala. Helps us with that. All right. Right near the amygdala, this picture doesn't show it because it's a little bit too anterior. But if we go back to this picture, excellent. And again, I wish your book had a better picture of this, but what we have here is this big, that doesn't show up at all. This big red blob. This big red blob, notice, also comes out laterally into your temporal lobe. And unfortunately, it isn't a big blob. What's actually really, really cool about, oh, I'm sorry, was that a question? All right. What's cool about it is this cluster of gray matter that has a big, huge body region. And this lump at the end is actually this really cool spiral of gray matter. So basically, it comes down and has this spiral of gray matter at the end here, which is actually where it gets its name. The name of this is the hippocampus. Anyone know what hippocampus means? Seahorse. Because its belly and the curl kind of looks like the belly and tail of a seahorse. The hippocampus is what is responsible for converting short-term memory into long-term memory. What do I mean by that?
Things that are happening day to day being stored into a memory that you can retrieve later on.
Yeah, absolutely. Excellent. That's great. And it doesn't even have to be day to day. I could give you three objects right now: ball, cat, fish. Repeat those back to me.
Ball, catfish.
There you go, ball, cat, fish. Excellent. So you have that short-term working memory where you were able to recall that information. Now, if I gave you a worksheet that had a bunch of really simple math questions like 5 plus 5, what's 5 plus 5?
10,
Or 7 minus 2?
5,
Right, or 2 plus 3?
5.
Right, and then I asked you for those three objects. What were they again?
Ball,
Ball, catfish.
Yeah, exactly, right? So you were able to maintain that within your working memory or store it and retrieve it as needed. Someone who has damage to their hippocampus can do the first part of that. I can give them those three objects and they're able to repeat them back because they're keeping them in their working memory. But then I give them a sheet of math questions. And by the time they've reached the end of that sheet of math questions, if I ask them to recall those three objects, they're not capable of doing it. There is a very, very famous patient known only as HM. HM is actually was a young gentleman who went camping. When he was camping, it started to rain. So he took his camping equipment, including his stove, and brought it into the station wagon with him while he slept. He ended up getting carbon monoxide poisoning, and he has bilateral damage to his hippocampus. And ever since that event, which now, gosh, is close to 40 years ago, he has not been able to make any new long-term memories. There are many neuroscientists that have worked with him. One of them was actually at Davis for a very long time. I don't know if he's still there, but he was. He was one of the first ones to work with him. And he worked with him for 25 years. And every day when he walked in to work with him, he had to introduce himself for the first time again. If he left to go and have lunch and came back again, he would have to introduce himself to him. Right? So 25 years and the guy never remembered him. The research that went on with him was truly remarkable, and it's taught us a lot about memory and working memory and muscle memory, that there really are different types of memory. Not surprisingly, HM has a tremendous amount of time on his hand. So one of the things he'll do is play video games. So here it is, Tuesday morning, you walk in, of course you have to introduce yourself to him. And then while you're in there, you'll ask him, hey, have you ever played this video game before? And he'll say, no, I've never played that video game before in my life. You put it in, you hand him the remote control, and he'll proceed to get the high score. And the whole time that he's playing and doing amazing in the game, he will be swearing to the top of the thrones that he has never played the game before in his life. And obviously, he is not able to make any new memories since that time. He is still able to recall some memories since then. But again, as you move 20, 30, 40 years away, right? How many of you remember things you were doing 40 years ago?
That would be hard because
Exactly. Most of you aren't 40 years old. Okay, well, how many of you remember things that you did when you were 3 or 2?
no.
Yeah, not a lot, whole heck of a lot. So, right. So yes, he has some normal memory loss from those events that have occurred, but he was able to retain some of it. Exactly, Stanley, you've hit it on the head. And obviously, he has some awareness that something has happened. If you think about it, 25 years later, when you wake up and look in yourself in the mirror, is the person looking back at you necessarily the person you would expect to see?
No.
No. So he has some understanding that something has happened to him. Now, notice also the hippocampus is very close to the amygdala. And his amygdalas were damaged in this as well. So his emotions in general are very muted. But so clearly when he looks in the mirror, he's aware that time has passed. He has a kind of a general idea that something has happened to him. But again, he's not able to really make any kind of meaningful long-term memories. And again, Samuel hit it on the head. There's not just one, but actually two really, really excellent and completely different movies made on this process. One, of course, is the comedy 50 First Dates, an excellent movie that does a really good job of showing this condition. The other one is a very serious drama. Anyone know the very serious drama that was made that was also equally excellent, involving
Finding
someone who
Nemo.
has... I'm sorry?
Finding Nemo.
Okay, three. I hadn't thought about that. Yes. So Dora in Finding Nemo is another example, but no, that wasn't actually the one. I'm not sure I would call that a drama. But anyone know the very famous drama?
Beautiful Mind.
Nope. Although that is definitely a psychological. No, Guy Pearce, Memento. In Memento.
A great movie.
Yes.
Great movie.
Exactly. Guy Pearce is a police officer who's a bad guy, breaks in, kills his wife, injures him, and he gets this hippocampal damage. So he's not able to produce long-term memories, but wants vengeance. So he has to take pictures and write notes and get tattoos all over his body to try to help him to solve the mystery and get the person who killed his wife. And it's a really amazing movie. One of the really interesting things about it is you watch the movie from his point of view. So it's kind of out of sync in time because of this deficiency of his. And in fact, when you first purchased the DVD, I don't know if it's still possible of doing this, but when it first came out on DVD, there was actually a special code you could put in that would actually allow you to watch the movie in linear fashion, as opposed to the way that you perceive it from his point of view. So again, two really, really excellent movies, all involving this damage to the hippocampus. All right, questions on that? Where do I want to go next? Let's do this. All right. Notice right down here, this little red dot that you see right here is known as the mammillary body. Now, is that on your list?
No.
So why am I mentioning it? Well, let me ask the question. Is it on your list?
Yeah, it's under the diencephalic.
Exactly. It is a diencephalic structure. So it is a structure that you are responsible for. It's not one of the cerebrum. It's not in the telencephalon, but it is one down here below the limbic system, and it is associated with the limbic system. So let's jump ahead a little bit and talk about it. The mammillary bodies, there's two on each side, one on each side, so two total, play an important role in taste and smell aversion. Right? Again, there you are out in the wilderness in ancient times, and there is a bush that has some red berries on it. You smell those red berries, you taste those red berries, and you get sick. And someone presents you with one of those red berries again, and you smell it. Are you going to want to eat it again?
No.
No. So yeah, bitter taste can do it, or getting sick can do it. Absolutely. Right. We want to avoid things that make us sick. And the mammillary body plays an important role in that taste and that smell aversion. Again, hopefully you're not wandering in the wilderness eating red berries, but maybe on a Friday night you had 10 too many tequilas. And now the smell of tequila makes you sick and queasy. Or my wife, when we were in Southern California, she was pregnant. There was a restaurant there that she loved to go to that had this really, really great peppercorn ranch dressing that she really, really liked. When pregnant one time, she ended up getting very sick, not because of the food, right? Just that morning sickness kind of thing that you know, nauseousness that you deal with pregnant. And she threw up shortly after eating a meal there. And did we ever go back to that restaurant? And did she ever have that peppercorn ranch ever again?
Nope.
Nope. Absolutely not, right? Exactly, because of that taste and smell aversion. And that mammillary body, it's a protective thing there to do that. And notice the reason I mention it now is because this taste and smell aversion obviously has to be related to our memory. And sure enough, in this three-dimensional view, we see that there is this bundle of axons that actually connects the mammillary body to the hippocampus. And this bundle of axons that connects them together is the fornix. Notice we see it really nicely here in our three-dimensional view. But what's also cool is in a sagittal section of the brain, we actually see this fornix as well. Here is the mammillary body. And here is that portion of that fornix. Notice the hippocampus is way over to the side, so we're not going to see that. But we do see it. And notice also our chart does a nice job of showing it as white because it is indeed a bundle of axons. Now notice it's not the biggest bundle of axons. The biggest bundle of axons is this big, huge comma-shaped structure known as the corpus callosum. The corpus callosum is our largest commissural tract. Remember, commissural tract is a bundle of axons that goes from one hemisphere to the next. We see it really nicely here in this sagittal section. But notice also, if we go back to our coronal section, here we see that corpus callosum as well. Notice we have our two hemispheres. Remember, this right here is our longitudinal fissure. And deep to the longitudinal fissure is this corpus callosum. Right. At the end of class, if you think about what happens at the end of class, you guys all stand up and give me that rousing, standing ovation. And that ability to be able to coordinate the movement of your two hands together, bringing your two hands together, obviously requires input from both sides of the brain. And so our corpus callosum helps us to do that. However, let's talk about something else that someone mentioned a few lectures ago. A few lectures ago, remember someone mentioned epilepsy. Epilepsy is a condition, as we talked about, where a cluster of neurons in a specific region of the brain become overactive. And they produce a big, huge, elaborate neural storm, basically where this huge, massive flow of action potentials spread not just across this hemisphere, but because of the corpus callosum, they can spread to the other hemisphere as well. When these big, huge neural storms occur, it can cause an epileptic seizure, where the person collapses to the ground and is violently convulsing, right? You have to put the spoon in their mouth to stop them from biting their tongue off. It is incredibly debilitating, right? Very hard for these people to live normal lives. They certainly can't drive a car, but even just sitting through a 2-hour movie can be problems. It's definitely sitting in a classroom for school is difficult and challenging, and it is an incredibly exhausting and debilitating and potentially damaging condition. Now, true, these tend to be localized in one area, so sometimes they could go in and maybe try to cut out the cortex, but again, cortex is important. We don't want to necessarily be removing that. So what's one of the ways that they can treat this condition?
I know there's medication for it, but I don't know what.
True, there are medications, but the medications don't always work. So in extreme cases, and again, I'll give you a hint, we were just talking about the corpus callosum, that commissural tract that connects the two hemispheres. So what do you think they do in some of these extreme cases? What are some of the things they can do with some of these people?
Got that in. Got the
corpus
corpus
callosum.
callosum.
Yeah, they actually will go in and cut the corpus callosum, right? Producing what they call a split brain. They will actually cut the corpus callosum. Notice if you cut that major tract, then what happens when that neural storm starts, it's basically not able to spread to the other hemisphere, dramatically decreasing the severity of that neural storm, and often dramatically decreasing the severity of the seizures associated with it. Now, of course, if we're cutting this corpus callosum, this major connection between the two hemispheres, then that must cause major deficiencies in the individual's functions, right?
I don't think so.
You would think so, but I'm asking the question, so what's the obvious answer?
No.
No. Shockingly, it is very, very hard. And there are some subtle cues, but it's actually these people act surprisingly normal. And again, one of the main reasons for that is the permanency of the world, right? There's a piece of paper here in front of me, whether I grab it with my right hand or my left hand, I can see where it is and I can find it in space. Often they have to produce these really elaborate tests to be able to test it. One of these cool tests, they'll put them in this viewer where they're shown a face. And it turns out it's an ambiguous face. One of the sides is male and one of the sides is female. And when they're looking through this special viewer at it, what they will tell them to do is they will ask them to write down what they're seeing, and they'll ask them to say what they're seeing. And they will write down male and say female. And the person won't be aware of the fact that there's a discrepancy there. Right? But like I said, this is a very specialized test. When you're walking through the world, how often is half of a person a man and half of a person a woman? Right? So surprisingly, they're really pretty normally functioning in the world in this split brain. So like I said, we see this corpus callosum, and again, a bundle of axons seen very nicely here in the sagittal section. But as we also talked about, we can see it really, really nicely. Coronal section. And in the sagittal section, not only can we see the corpus callosum, but we can also see the fornix as well. Now notice in between the corpus callosum and the fornix is this thin membrane. Again, notice if we go back to our frontal section, underneath the corpus callosum is a thin membrane. This thin membrane is a structure known as the septum pellucidum. If you remember, when we were talking about cerebral spinal fluid, we mentioned how cerebral spinal fluid surrounds the brain, but there are also some spaces inside of the brain. Two of these spaces are these big, huge openings right here, known as the lateral ventricles. And I think I have a really cool picture that shows this. There it is. Notice now we are looking down in this kind of weird transverse section, multi-leveled transverse section. Notice we can see a little bit of the corpus callosum, that fiber tract that crosses the two hemispheres, that big comma shape that would be sitting there on top. But notice also here around some of these nuclei, we have what turns out to be this big C or horn-shaped space. And these two big horn-shaped spaces
are known as the lateral ventricles. And notice the lateral ventricles do not meet. There's a little bit of membrane in between them, separating them from each other. And that is the septum pellucidum. So if you think about what's going on in that sagittal section, oh, I like this, let's do this. In that sagittal section, notice when we're looking at it with the sagittal section, we are just looking at one side of it, where we see that membrane between the corpus callosum and the fornix. And so if you think about it, if we were to take a probe and poke through that septum pellucidum, we would be in the lateral ventricle. So if we go back to this picture, this one works. Fornix, corpus callosum. And if we were to poke that septum pellucidum membrane, we would go right into the lateral ventricle on the right-hand side in the right hemisphere of our cerebrum. All right. Questions on that? All right. Two more big things we need to talk about. No, where is it? Okay, we'll come back to this. No. Oh, no, that's not what I want. I want this picture. Excellent. Okay. Notice, and again, this is something we have talked about before. Underneath the frontal lobe is an enlarged stalk, a large bulb with a stalk coming off of it. Notice here the close-up view, enlarged bulb and the stalk coming off of it. This is the olfactory bulb and the olfactory tract. The olfactory bulb is actually a cluster where we find cell bodies, so it's actually a nucleus. And then their axons go back to the limbic system. And what is it that our olfactory bulb and our olfactory tract provide us information on?
Smell.
Yeah, okay. I know you guys are getting tired. I know it's getting late. How are we on time? We're doing good on time. I know it's been a long day, but we've got nothing but long days ahead of us. So try to hang in there. Sense of smell, absolutely. Remember, we talked about this once before when we were looking at the bones. Remember here, we saw that olfactory bulb sitting on top of the cribriform plate, where we said then the olfactory nerves come into the nasal cavity to provide us with that sense of smell. Well, what's interesting is the olfactory bulb connects to the olfactory tract, which carries that information back to the limbic system. All right, it goes to the limbic system. And again, you may not have thought of it in these terms, but you are 100% well aware of it. Because which of your five senses do we most closely associate with memory and emotion?
Smell.
Smell, absolutely, right? When I'm missing that special someone, I'll call up my phone and listen to the last voice message he left me. I'll look at the pictures that we took of that trip we took together to Boca. But when I'm really, really feeling sad and gloomy, I will bring out that Ziploc bag that has his shirt in it. And I'll open it up and take that little smell of him and then quickly close it back up again. And that smell of his musk from that shirt brings back all the emotions and all the memories of all that wonderful time we spent together. Right? Our sense of smell, that smell of a fresh baked pie reminds you of that summer you spent at grandmother's house.
all right, swinging on the swings and swimming in the pond and all of those. Our sense of smell is the one that is most closely associated with our emotions and our memories. And notice that also brings it to the mammillary body as well to help us to protect us from anything harmful that we may smell as well. I think I told the story about my sister's boyfriend and the olfactory bulb getting drunk, falling down the stairs. Do we do that? Okay. No, yes. My sister dated a young gentleman, and one New Year's Eve, he got incredibly drunk and fell down a flight of stairs. And as he was bounding down the stairs, and his brain was rattling around in his skull, his olfactory bulb ripped off his cribriform plate. And when he woke up that morning and every day since then, what has he not been able to do?
No.
Yeah, he lost his sense of smell. Exactly. But as I've said many times, it has a very happy ending. She broke up with him. All right, so that is our olfactory bulb. That leaves us with one last structure, and let's go back here. Fun with vocabulary. Here in deep within our limbic system, within our cerebrum, there are these 3 nuclei. These nuclei are kind of like a muscle group. They have their own individual names, the caudate, the putamen, and the globus pallidus, but you're not going to need to know their individual names. You just need to know that collectively they are referred to as the basal ganglia. And again, let's think about that term. We know a ganglia is a cluster of cell bodies in the peripheral nervous system. So this has been horribly, horribly misnamed. Thankfully, more and more people are starting to refer to it as the basal nuclei, which is a much more appropriate name, but you will still see basal ganglia in the literature. So I will accept either name for these. So again, like a muscle group, you don't need to know the individual names. You just need to know these things collectively. And collectively and appropriately, they're known as the basal nuclei. Although you still will sometimes see them referred to as the basal ganglia, and I will accept that. All right, now that we know what they are, let's talk about what they do. Their job is basically to maintain, let's say it this way. They maintain subconscious control of skeletal muscle. That's fun to say. Again, the kind of thing you can call grandma on the phone and tell her, hey, grandma, guess what? Your basal nuclei maintain subconscious control of skeletal muscle. But what the heck does that mean? Well, in simple terms, right, this is your autopilot. What does your autopilot do?
Keeps just kind of active, keeping the muscles active and maintained for structure and support.
Exactly. This helps us to maintain a learned behavior, a learned action. If you think about a 1-year-old when they're first learning to walk, you can almost see the steam coming off the top of their head as they're thinking so much about where their arms are going and where their feet are going and where their body is going as they're trying to learn how to maneuver themselves through space. However, now you walk while drinking a cup of coffee and eating a sandwich and tweeting and doing all sorts of other things. Driving is a great example of that, right? Knitting. Knitting is one of my favorite examples. My grandmother tried to teach my wife's grandmother tried to teach my wife to knit, right? And again, while my wife's sitting there trying to do each purl and each knit, right, she's giving it her complete focus. Does grandma need to give her knitting her complete focus?
No.
No, she's whipping out stockings while she's talking to you and watching TV and having a conversation and smoking a pipe and all the other things that grandma does. Notice it maintains. It doesn't initiate it. You don't just spontaneously start driving or spontaneously stop driving. But when you're driving, your autopilot kicks in, which is both a good and a bad thing, right? Because yes, it maintains that driving behavior. But if you get too much into your feels, then suddenly you're halfway to work and realizing, oh, you were supposed to be going to the store. Or you're standing there in the shower trying to remember whether or not you washed your hair because your autopilot kicked in and you can't remember whether or not you washed your hair or not. You remember getting it wet, but then you got daydreaming about anatomy and physiology and you can't remember whether you actually washed it or not. So these basal ganglia, these basal nuclei, help us to maintain learned skeletal muscle behavior. Again, at a subconscious level. If you're passed out, you're not going to spontaneously start driving to work. But once you initiate a learned behavior, this helps to maintain that activity.
Did that have any effect on like sleepwalking?
Potentially, right? So again, one of the things that happens when we're asleep is that our brain shuts off the motor function to things. So that when you're running or jumping or throwing things while you're asleep, your body doesn't actually move to those actions. However, there are some people who can act out those, and I'm not fully familiar with the cause of that. My guess is that it might be more cerebellar than the basal ganglia, but definitely we're more likely to do learned behaviors, walking, things along those lines. So potentially it could be involved in that, sure. All right. Questions on that? All righty. With that, we are done with everything we need to talk about for the, I did that. Telencephalon. As you can see, that is the majority of the heavy lifting. We do still have the protections of the brain, but I think I want to talk about the spinal cord before we get to that, at least a little bit. So from here, we are on into the diencephalon, mesencephalon, metencephalon, and myencephalon. But I think I have tortured you enough for today. So I think even though it's a little bit early, it's almost noon, I think that's some lot of heavy lifting. We're kind of almost caught back up to where I'd like to be. So I think this is a good stopping point. So I think we can go ahead and stop here. What we are going to do on Thursday when we come back is we are going to pick up right here, starting with our lab stuff first, and then finish catching up on our lab. And then from there, we'll go into our lecture on the spinal cord. We've done the brain, but we're not quite done with our central nervous system. There's some spinal cord stuff we have to talk about for that. And then we'll finish things off the following week with, like I said, our autonomic nervous system. All right. Questions on any of that? All right. Again, this is one of your one-day weekends to study this material. You have seen how we play the game, going through this list, identifying structures, identifying functional regions, and if an area has a function, you are responsible for what that function is. Right? Notice the septum pellucidum is basically just a structure. It's a dam between the two ventricles, so it doesn't really have a function, whereas obviously the amygdala has a specific function. So those are the things that are going to be on this test. Identify the structure, identify this region, identify what that region does. That's what the majority of this test is going to be. All right. Questions on any of that? All righty. I will, before I get off, I will go ahead and stop then, and let's go ahead and stop recording.